بسم الله الرحمن الرحيم. السلام عليكم جميعا، أسعد الله صباحكم بكل خير. معكم دكتورة مها العماري، منسقة يوم الصيدلي للأبحاث. يشرفنا حضوركم وتواجدكم معنا في هذا اليوم. الزملاء والزميلات الأفاضل، يشرفنا في هذا اليوم إعلان انطلاق يوم الصيدلة للأبحاث في مدينة الملك عبد العزيز الطبية بوزارة الحرس الوطني بالرياض. هذه المنشأة والمتمثلة في قيادتها الحكيمة والتي دأبت دوما على الحرص على التميز في جميع المجالات، حرصت أيضا على تقديم أفضل الخدمات والرعاية الصحية المتميزة لمرضاها، والتي تعتمد بالمقام الأول على السلامة الدوائية، وذلك من خلال دعم قسم خدمات الرعاية الصيدلية في شتى المجالات، والتي تعتبر الأبحاث الصيدلانية أحدها وأهمها أيضا. لقد أثمر التعاون المستمر ما بين الخدمات الصيدلية ومركز الملك عبدالله العالمي للأبحاث الطبية وكلية الصيدلة بجامعة الملك سعود بن عبد العزيز للعلوم الصحية عن مخرجات ترقى إلى مصاف الدول العالمية المتقدمة في الأبحاث العلمية. إن الأوراق العلمية التي نتجت عن هذا التعاون المثمر تم نشرها في مجلات عالمية ومرموقة، ولا يخفى على الجميع ما تتطلبه الرحلة البحثية للصيدلي من عمل دؤوب ومتابعة مستمرة، بالإضافة للحرص على الالتزام بالأخلاقيات البحثية والأمانة العلمية، مع التركيز على الرؤية المستقبلية للمملكة العربية السعودية. ولله الحمد، هذا ما بدأنا تحقيقه بفضل من الله، ثم بفضل الجهود المميزة لكل من ساهم في إخراج هذا اليوم، والذي نطمح أن يؤتي ثماره المرجوة ذات الأثر العميق على جميع القطاعات الصيدلانية وعلى تطور مهنة الصيدلة بالمجمل. هذا الملتقى لم يكن ليصل لهذه المرحلة لولا الدعم الكامل من إدارة قسم الخدمات الصيدلية، متمثلة في الدكتور سعد العبيدي والدكتور صالح الدخيل والدكتور خليل الماجد، ولن أغفل روح الفريق العالي اللجنة المنظمة والعلمية، والمكونة من الدكتورة نوف الحارثي، الدكتورة دلال عبد الكريم، دكتور خالد السليمان، دكتور عبد الرحمن التراكي، دكتور عبير السماري، دكتور خليفة الذياب، دكتور نعمان العبدان، ودكتور أشجان الغانم، والدكتور ماجد النهاري، والسكرتير مايكل منابت أيضا، وأيضا التعامل المهني العالي للأستاذ فريان الفريان، مركز الدراسات العليا بالحرس الوطني. كما أتقدم بخالص الشكر والتقدير لجميع المتحدثين البارزين والباحثين وأعضاء لجنة التحكيم الذين لم يترددوا للحظة في التعاون معنا لإنجاح يوم البحث الصيدلي الافتراضي الأول بمدينة الملك عبد العزيز الطبية بالحرس الوطني، راجين من الله العلي القدير التوفيق للجميع خلال هذا الملتقى والحصول على الفائدة المرجوة منه. بسم الله الرحمن الرحيم، نبدأ يومنا إن شاء الله. Our first speaker today is Dr. Majid Al-Jraisi. Dr. Majid Al-Jraisi is a well-known figure at National Guard Health Affairs. He obtained his bachelor and master degree in clinical pharmacy from King Saud University in years 1991 and 1996, respectively. In 1998, he obtained his Doctor of Pharmacy degree from University of Tennessee at the United States, followed by 2 years of residency and fellowship in pediatric pharmacotherapy. Dr. Al-Jraisi has also completed a Master of Science degree in Medical Education from King Saud bin Abdulaziz University for Health Specialties in 2009. Dr. Al-Jraisi has authored and co-authored numerous and relevant scientific research projects that were published in reputable journals. Currently, Dr. Al-Jraisi is working as a pediatric clinical pharmacy consultant and associate professor of pharmacy practice, College of Pharmacy at King Saud bin Abdulaziz University for Health Science. He is also a chairman of Research Office and director of Clinical Trial Services at King Abdullah International Medical Research Center. Dr. Majid Al-Jraisi will present his speech now. Dr. Majid, the floor is yours.
Assalamu alaikum wa rahmatullahi wa barakatuh. Thank you very much, Dr. Maha. Just to make sure that my slides are clear and my voice is clear as well. Can you hear me?
Yes.
السلام عليكم ورحمة الله وبركاته. صباحكم الله بالخير جميعا. في البداية أشكر الدكتورة مها العماري على تنظيم هذا الحدث المهم جدا جدا، ليس فقط على مستوى الشؤون الصحية بوزارة الحرس الوطني بل على مستوى المهنة في المملكة. أعتقد أنه يوم مهم جدا في our pharmacy profession ويعني راح يكون إن شاء الله strategic goal for our research, pharmacy research. My talk is going to be about the National Command Center for Clinical Trials, Opportunities for Pharmacists. Let me start with the basic definitions, and I'm sorry for this, but just to set the ground on the definition of clinical trials based on the International Committee on Harmonization for Good Clinical Practice. It's any investigational in human subjects intended to discover or verify the clinical, pharmacological, and/or other pharmacodynamic effects of an investigational product, and/or to identify any adverse reactions to an investigational product, and/or to study absorption, distribution, metabolism, and excretion of an investigational product with the object of ascertaining its safety and/or efficacy. So it is not only for investigational drug, but also include medical devices as well, because it is an intervention and it is considered as there is a safety and efficacy also for devices. The terms of clinical trial and clinical study are interchangeable and synonymous, so it can be used both in both ways. So where is the position of clinical trials in the journey of the research and development innovation, where the basic research start from research idea and then testing the research idea and doing it on the bench side, and then moving it into preclinical, which is conducting this study on animals just to test the safety, and then it goes to the phases or clinical trials, which is the 3 phases that we all know, phase 1, 2, and 3, and then moving into, which is important, to the medical products development and commercialization, where if it's drug to be marketed or vaccine, or it could be a medical device. The clinical trial team, as we all know, it is composed of many members. So the clinical trials facilities are mixed environment with individuals from different background, having variety of skills, knowledge, working on research health site, equipped with patients and hospital staff and system. And the member of clinical trials, they are including but not limited to physicians, nurses, pharmacists, laboratory personnel, other allied health sciences like respiratory therapist, dietitian, occupational therapist, research coordinator, project manager, legal advisor, financial advisor, and data analyzer. So you can see that clinical trial is a team collaborative approach. It's not one-man show. It cannot be done by only the investigator, but it has to be with different members because it is a complex process. Why we are keen about conducting clinical trials in our country? There are many reasons. First, participate with the developed countries in the scientific knowledge and experience, and join publications in high-impact journals. So we have to be part of that involvement in the scientific and participate in the scientific knowledge and community. Enhance the capabilities of the healthcare providers to do high-quality clinical research studies. Establishing and enhancing the infrastructure of research sites to conduct clinical trials, providing clinical data from Saudi community to compare it with the other international participants for assurance of safety and efficacy, or if there is any ethnicity differences between us and the other countries. Facilitate the availability of alternative therapies for complicated and rare diseases, and this is very important. Accessing the in early phases of these medications, especially if it's very expensive medications. Nowadays, we see the monoclonal antibodies, very, very expensive medication. Getting access to these, it's not only the cost, but also the access, because if the drug is being marketed, we probably we will not have access to that medication, investigational drug, before a few years launched in the original country. So this is opportunity for our patients with complex diseases, oncology patients, rare diseases, to have access, autoimmune diseases as well, to have access to this investigational drug early on. As a good example for a medication that is used for the spinal muscular dystrophy, cost $500,000. So this is having access to that medication early on is advantage. It is considered as one of the sources of generating revenues and high economic impact in opening positions for the Saudi employees. And again, we will talk about this in a few minutes, but really this is important for generating revenues, especially we're talking about 2030 vision with economic impact on the country. The sponsor, the pharma sponsor, they are very meticulous about selecting the countries and the sites. So based on what they are selecting countries and sites, it's based on the following. First, availability of experienced clinical trial sites. And they are looking for at least more than 5 sites in each country because the process itself is very complicated. So they don't want to invest the time for only one site. Availability of patients, of course, and this is during the feasibility study. Availability of clinical trial infrastructure and sites. Standards of care similar to the North America and Europe. Short and predictable startup timelines because they don't want to be behind in recruiting patients. Well-functioning and predictable regulatory environment. Fortunately, we have a very robust and good system with our regulatory body here, the Saudi FDA. History of previous successful participation in different trials because they want to have a track of records for conducting high quality research because the first impression is the last impression. So if we participate in any clinical trial with high impact, this will reflect the future involvement of clinical trials. The competitive per patient cost more than just investigator and hospital fees. This is something that is high opportunity for our country, for our region, in fact, to compete with others, especially the cost for conducting clinical trials in Europe and North America is increasing. So coming to our country and to our region, in fact, is advantage. No unusual country requirements. Governmental support for clinical trials, and this is the stick and carrot. So the government has to support this with logistics, with facilitate the conduct of clinical trials in all sites. Importance of countries market if the product is launched. Again, this is something that the pharma company looking for. So if there is a potential for the investigational drug to be launched, to be marketed in the country, then it is an opportunity to have the study conducted in our country. What are the contract research organization, which is the CRO? This is contract organization by the pharma company. They can influence, in fact, the selection of the sites based on the following: availability of clinical trial department, single institutional level point of contact, because they are the one contacting the clinical sites for conducting clinical trial, willingness of investigators to conduct clinical trials and to do feasibility study, institutional awareness support for clinical trials, well-functioning IRB committee, availability of required equipment, simple contracting procedure, and past performance study startup predictability, productivity and quality of easy to communicate with. So this is the role of CRO, and as you know, there is a sponsor company directly communicate with the sites, or there is contract research organization communicating with the site to conduct the clinical trial. This is just to give you a hint on what are the priority for the clinical trial, the CRO in selection the countries. So here, as they get closer to the outer circle, it is more priority to them. So the top priority is approval time, which is the timeline and access to patients, and then it comes the quality and the expertise comes next, and then the cost comes third. So these are the priority for selection of the countries. Now, moving from the requirement to conduct clinical trial into the need for clinical trials globally. So globally, there is an increase of 19% in the past 2 years of conducting clinical trials. Let's just take an example. In Europe, for instance, Germany, UK, France, and Italy, the 2 years average growth rate for clinical trials, 15%. When it comes to the North America, the average growth curve is 13%. In the African countries, it is only 20%. Now let's go to our region. In our region, most significant clinical trial, Egypt and Saudi Arabia, the growth rate is 34%. So you can see the opportunity for our country compared to the Korea, China, and Japan is 33. So they are our competitors, in fact. So maybe if they have a good infrastructure in clinical trials, they have already well-established expertise to conduct clinical trials, the sponsoring company will go to that part of the world and not coming to our region. And the last one is the India, which is the growth rate is 27%. Now let's take the clinical trials from different perspectives, which is the pharmaceutical spendings. Now looking at from the left side, the countries, of course, the US is the largest country conducting clinical trials, followed by the Europe and also the Japan, and then the rest of the world. When it comes to what type of clinical trials, original brands, product, comes first, 52%. Then next is the non-original brands, which is probably the generics, and then the unbranded and the OTC becomes the equal. When it comes to which area of clinical trials, the sponsoring company now moving towards is first and the largest is the non-communicable diseases. And non-communicable diseases meaning the cardiovascular, the diabetes, and I think everybody knows that unfortunately our country, one of the largest prevalence diseases of non-communicable diseases we have it in our country. So this is, again, it is unfortunate, but at the same time, let's take it as an opportunity to recruit more clinical trials to our country. And then comes the oncology, and then diabetes is as well in the 15%. So this is where the clinical trials moving to towards in terms of the type of diseases. Now, Saudi Arabia is the highest in medication spending per capita, which means how much we spend per person in terms of the spending of pharmaceuticals per person in our country. You see here we are the largest, the highest spending. This is unfortunate. We are not proud of this, but it reached $300 per capita. So we are spending 300 or around 1,100 riyals per person for pharmaceuticals. And the next is Poland, next to us, which is again about 29%. It's less than 250. So again, as a proportion to this, we spend more in pharmaceuticals. We should be conducting more of clinical trials. This slide is this data from the Ministry of Health data, and this is the latest data, just to show you how much we are importing pharmaceutical products, 70% importing compared to only 30% local manufacturing. So again, this is an opportunity that for the brands that we are importing or generic we are importing, we should be conducting more clinical trials to offset the expenses of this pharmaceutics, at least that is very expensive. In medical devices, 98% imported and only 2%. So we need to move in the direction of the innovation and patents. With regards to the accessibility of clinical industry, sponsored phase 2 and 3 clinical trials, the red one considered as the highest and the green one, the light green or the dark green, considered these the lowest, as you can see here from the chart. So in the North American region, they are the highest in conducting clinical trials. Also in Australia, it becomes also a high demanding country for clinical trials. Also in Europe as well. But in our region, we are still behind in terms of conducting clinical trials by the sponsor in phase 2 and 3. With regards to the benchmarking, let's just take an example of benchmarking, and this is an example of Poland. Poland is a country similar to our size of the country, so their population 38 million people. We are 33 million people. Our GDP approximately 782 billion. They are 585. But look at their clinical trials. It's approximately reaching 6,296 clinical trials compared to only 445 clinical trials in our country. And you look at the growth in the upper right corner is in 2019. You see the Poland, how they are growing, while in our case is very slow growing. And their 2020 horizon, their biotech is provision of $700 million. In our case, the vision is not clear on terms of the biotech. Here is the estimated, as I mentioned, the revenues from clinical trials is estimated in our country as the global percentage of global of clinical trials in 2014 is 0.022. With that involvement and spending, the expected clinical trial revenues from only this percentage is $180 million from the clinical trial. So imagine this is data of 2014. So with this data of 2020, imagine that this number will be tripled if we conduct clinical trials. So again, it is generating high revenues for the country, as well as opening opportunities and positions for the Saudi employees. Now, in terms of the expenses and the cost for clinical trials, this is just an example of US phases. And here is the dark one is the phase 1, the light blue is the phase 2, the green is phase 3, and the last one is the phase 4. So you can see the trends of increasing phases of clinical trials. So here in the 2020 is going up, 2021 is expected to increase more. So the direction of clinical trials in different phases are moving up. So we have to be moving with this direction. Now, with regards to the type of the clinical trials, this is data of 2018. Autoimmune diseases represent a high percentage compared to oncology probably the largest one, and also CNS condition, as well as the diabetes. So these are the main areas that if we want to focus on, we need to focus on these areas for spending more time and more efforts in concentrating on clinical trials in those areas, because it is the future of sponsoring company to invest more. And if we are talking about the cost, this is just to give you an example of how much it costs per phase, per patient, by the way, in different phases. So if we take an example of infectious disease clinical trials, in phase 1, it costs per person $15,000 per person. Phase 2, $17,000, phase 3, $18,000. And you can go on and on. For the oncology, look at the costs, $57,000 per patient for phase 1 and going to $69,000 per patient in phase 3. So again, it is considered as very expensive, very good source for revenues for the country. Now, in what expenses, in what areas that these clinical trials will be spending on? First, the type of cost that's per patient, of course. So look at the elements. So patient recruitment cost, patient retention cost, registered nurse and clinical research associate cost, physician cost, pharmacist cost. And when we say pharmacist, it's not only preparing, it's from logistic and shipment into the involvement of clinical. We'll talk about the role of pharmacists in this, into dispensing and preparing, and also supervising the conduct of a clinical trial in terms of randomization and concealment. Clinical procedure cost, central lab cost, and per site. There are also listed costs in each category. So this is per patient, per site, and per study. Now, are we ready for the clinical trials to be a hub of our clinical trials? These are the opportunity for our country. So location and population, geographical location, facilities, communication and interaction. We have a good reason to be the hub for clinical trials. Healthcare infrastructure, I think most of our infrastructure for health facilities are very advanced, and it is really advantage to attract the sponsors and the CRO for conducting clinical trials. Education, we are highly educated. Our universities are producing high level of graduates, high quality of graduates, including pharmacy colleges. So we should be again ready for conducting clinical trials and also be attractive for the sponsor company and the CROs. In addition to these characteristics, the cross-sector collaboration. Again, it is we have the long-term national science and policy and technology and policy in place, so which is again gives us an advantage. Governmental support and structures, again, it is improving. Of course, we are not saying it is ultimate now, but it is improving for the infrastructure. Now, let's talk about Saudi National Command Center for Clinical Trials. The Saudi Clinical Trials Enterprise, this is the name of this National Command Center. And this is according to launched as part of the National Vision 2030. Sponsored by the National Industry Development and Logistics Program, charged to develop and enhance the capabilities and the capacities of clinical trials in the country, with the ultimate goal to contribute effectively and the development of R&D pharmaceutical industry in the Kingdom of Saudi Arabia. As we mentioned, that somebody has to take the lead and has to organize all these efforts in order to become attractive for the sites. Now, the milestones for the Saudi Clinical Trial Enterprise, first signed the MOU between KMARC and Connect, our strategic partner for establishing the National Command Center. It was the fourth quarter in 2017, then followed by the visit from Connect to our facility in the first quarter of 2018. Then later on, the Korean team, they came and conduct training in our site in the second quarter of 2018. Then later on, the phase 1 launched of the phase 1 clinical trial was launched in the fourth quarter of 2019. The approval of the NIDLIB for our strategic plan was the second quarter of 2019, and formed the Saudi Clinical Trial Enterprise formally with advisory board in the fourth quarter of 2020, and we had our first meeting. So this is the signing ceremony with the Korean partners. It was in June 2019 with the presence of our CEO and the CEO of the Seoul National University in Korea. These are the members of the first advisory board, the Saudi Health Council, King Fahd Medical City, Connect, the Saudi Food and Drug Authority, King Faisal Specialist Hospitals, and there are other centers are going to be also included in the advisory board. What is the key strategic objectives in the National Command Center? First, establishing a national network to help manage and coordinate clinical trials and act as a key link among national stakeholders and international partners. As I mentioned, somebody has to take the leadership role in order to coordinate and to collaborate with these centers. Establishing effective strategic partners with several internal and external academic, governmental, and industrial institutions running and or sponsoring clinical trials. Again, when the sponsors or the some institutional universities looking for sites to conduct clinical trials, we can be the representative for the country in order to communicate with these sites and also the sponsor. Supporting economic diversification by providing an attractive investment environment for local and international investors. One of the key strategy for Korean, by the way, I didn't talk about why we have chosen Connect as our partner, because the steps that they went through from during their journey of 15 years, they were in the probably in the infancy period 15 years ago, and now they are the one of the top 5 countries worldwide conducting phase 1 clinical trials. One of the strategic key, success key, is they have recruited, they have went on and talked to the sponsors, and they have recruited clinical trials to their country and start to build the infrastructure of their sites in order to be ready for clinical trials. Promoting medical research by raising the level of competence and of practitioners in the kingdom. This is one of the main key strategic goal for us, is to enhance the capability, the skills, the knowledge, the expertise of the clinical trial members to conduct high quality, because the sponsors are looking for high quality data. So again, they are not ready to compromise the integrity of their data because one of the sites are not capable of conducting clinical trials. Providing opportunity for capacity building and training in clinical trials in all sites across the country. Now, let me just give you a background of where do we stand for now in terms of clinical trials in KMARC. How much time I have now? How much time I have? Can you hear me?
Dr. Majid?
Yes, how much time I have?
You still have 15 minutes.
Great, thank you. So, where do we stand for now in terms of clinical trials? This is just an example in KMARC because we know our structure and we are not yet developing the data for other sites. So we have established the clinical trial services in KMARC to be able to coordinate and to conduct clinical trials. So this clinical trial services that we have compose of multidisciplinary team that is highly experienced in conducting clinical trials in a variety of different diseases. Each clinical trial that we have recruited or we are part of, we provide a research coordinator. This research coordinator is playing a vital role in the success of any clinical trial. Without having a research coordinator in the clinical trial, I don't think the clinical trial will be successful, because the role of clinical research coordinator in any clinical trial is vital, is the link between the sponsor, the investigators, the principal investigators, the site, and also linking and communicating with all team members for the clinical trial. So it is not the principal investigator. Of course, the principal investigator is the responsible person, but somebody has to be the link, the liaison between all different parties in any clinical trial. So in this services, we provide scientific offers, study design, protocol development, feasibility studies, and biostatistical consultation for clinical trial. In case of the investigational clinical trial started by principal investigator. Project implementation, site startup, site management, patient recruitment, investigational drug management. And we are lucky enough to have investigational drug unit that is that have highly experienced investigational drug pharmacist and also pharmacy technician. It provides huge help and support to investigators, as well as to communicate with the sponsor during the early communication for clinical trials. In fact, the CRO, when they come to assess the site for feasibility, one of the main areas that they had to visit is the pharmacy department. In pharmacy department, they need to make sure that they have logistic in place, they know how to handle and store, and also process the investigational drug. So we are lucky enough to have a good service in this part. Project management, assignment of clinical coordinators, as I mentioned, training of clinical trials on the clinical trial conduct, site monitoring. We also provide research monitors. As far as I know, we are the only site who have research monitor among all other sites. And this is something that we need to do and help other sites to develop their human capacity to be able to monitor their own clinical trials. Data management, we have again a good data management section in our facility in KMARC, providing and helping to create the case report form, electronic case report form. And one of the successful stories that we have is, and which is lately published in High Impact Journal, is our miracle trial, which is the MERS-CoV clinical trial, led by Professor Yasin Arabi. We have many sites, participating sites in this site, sponsored by KMARC, this site, and producing high quality data, which is again indicate the capability, or our capability of conducting high quality clinical trial that deserve to be published in New England Journal of Medicine. Safety reporting, we have a good system for safety detection. It has become part of the safety reporting, the SRS system in our facility, in our institution. Archiving and quality assurance as well. This is just a picture of the first phase 1 clinical trial unit that was launched in, as I mentioned, in the late 2019, that was approved by the Saudi FDA. This unit is located in Unit 72 in Cash building in the 8th floor. I would highly encourage you to come and visit this unit. It is the only phase 1 clinical trial unit, by the way, approved in the country, and we are working hard in fact to expand this. This unit is capable of conducting phase 1 clinical trial as well as bioequivalence studies that hopefully, inshallah, we will be starting soon to conduct bioequivalence studies. This is the facility of this phase 1 unit that include the recreation center for the volunteers. These are the beds, the rooms for the volunteers, as well as the area, waiting area for the volunteers, and the reception area. In terms of how many clinical trials we have in KMARC, we have a good number of clinical trials, and again, we are proud to be the probably largest site in the country to have this number of clinical trials. Again, our clinical trials mainly are distributed among oncology, second by the infectious disease and the cardiovascular as well. So we are conducting a very high quality clinical trials, sponsored as well as investigator-initiated clinical trial. So what is the ultimate goal? Where are we going? Which direction for this national command center? So we start with building clinical trials infrastructure in our capacity, in our sites, as well as the sites in other countries in different regions of the country. And then to deliver high quality clinical trials conduct. As I mentioned, the clinical trial, the sponsor are looking for high integrity, high quality data. So we have to maintain the high quality data generated from the clinical trials, become the hub for clinical trial in the region, and this is what we are looking for. If you remember, we have 34% growth rate of clinical trials. We should be moving into that direction in order to be the main focus of clinical trials in the country. We have competitors, as I mentioned, Japan, Korea, and China, so we have to be competing with their sites. Now, the last part in my talk is going to be about the opportunities for pharmacists. So what is the opportunity for pharmacists in clinical trials? As you all know, the role of pharmacists in clinical trials, we have different roles. As a clinical pharmacist, as a staff pharmacist, inpatient and outpatient, investigation and drug pharmacist, administrator pharmacist, pharmacy technician. They are all playing major role in the conduct of clinical trials. We cannot say one pharmacist can do all the job. It cannot be. As I mentioned, we are proud, in fact, of being qualified site. In many cases, I can say with confidence, 90% of the feasibility visits conducted by the sponsor or the CRO that coming to our site, when they visit our site to assess the capability of pharmacy infrastructure for conducting clinical trials, we always pass. We are really having a good team, and thank you very much for the support from the pharmacy administration in conducting and continuing providing high quality services to the clinical trials. So where are the involvement of pharmacists in clinical trials? Let's start with the pre-clinical trial, reviewing clinical trial. This is just an example, by the way. It's not, I didn't include all the rules, but this is just to give you some glimpse and some idea about the role of pharmacists in clinical trials. So pre-conducting clinical trial, reviewing clinical trial protocol, checking for randomization process. This is very important. The only person that can be unblinded, that can have successful randomization is the pharmacist.
If you don't mind, we need to wrap up for the opening ceremony.
Logistic, sure. Logistic arrangement, drug shipping and handling, coordinating with pharmacy sections. During clinical trial, responsible for the ID dispensing, the investigation drug dispensing, maintaining concealment, and preparing the investigation drug. After the clinical trial, it is attending clinical trial investigator meeting, supervising the unblinded process, reviewing the manuscript for publication. So my conclusion is, clinical trials are considered as one of the key performance indicators for any country to develop their infrastructure and to be, in fact, from the top countries in conducting clinical trials. There is a remarkable increase in the global demand for clinical trials, and we have to be at that level of the high demand and catch this opportunity to have our country as the hub of clinical trials. Our country has very high potential to be the main site, the main country, the main region for conducting clinical trials. The role of pharmacists in the conduct of clinical trials is very essential and vital for approving the site and approved the country to conduct clinical trials. With that, I would like to thank you all for your listening.
Thank you very much, Dr. Jeresi, for this excellent talk. thought that we really needed this time in the era of clinical trials in the kingdom. Is there any question from our guest speakers? If you will allow me, I'll ask you this question, and if you can shed a light on the reasons why the Kingdom of Saudi Arabia has a very high growth rate compared to other countries, 34%. What do you think are the reasons for this, Dr. Majid?
Well, this is a very good question, Dr. Maha. In fact, the sponsor company and the CRO as well, they did their assessment, feasibility assessment, and they are looking for which country that has emerging markets. So there are many reasons. One, as I mentioned, the spending pharmaceutical expenditure in our country is very high. So we are emerging country for spending more pharmaceuticals, and which is likely to have launched their pharmaceutical product. This is one. Second is the not the low cost, but the acceptable cost for running clinical trials, because alhamdulillah, we are a high income country that we can minimize the cost that is really paying for the clinical trials team compared to Europe and North America. So this is another attractive that we have to keep in mind. Third reason is the infrastructure of our country compared to other countries, neighborhood countries, for the facilities, healthcare facilities, as well as the system that we have, electronic system. This is the top 5 reasons for being in the high probably market for clinical trials among other regions.
Excellent. This is very excellent. Dr. Majid, is there a very clear rule for all pharmacist level and the clinical trials among the connection that you presented? I mean, a rule for the pharmacist, a rule for clinical pharmacists, for the administration even. Is there a very clear defined rule for every one of them?
This is something that we need to work on. We are trying to promote, definitely, of course, as you all know, conducting clinical trials without involving pharmacists right now is impossible. So in terms of coordinating with the sites, coordinating, I mean, with the pharmacy, different pharmacy sections, inpatient, outpatient, pharmacy store, and also dispensing of medication. So there is a rule, of course. And by the way, who is requesting the rule is the sponsor themselves or the CRO requesting to have a pharmacist, designated pharmacist for the clinical trial. So without nominating and designating pharmacists for the clinical trial, they will not approve the site.
Excellent. We have another question from Dr. Khalifa Diab, Dr. Majid, if you will allow us.
Assalamu alaikum, Doctor. Thank you so much for your presentation. Doctor, comparing with other specialties, and are we behind in the clinical trials?
What do you mean other specialties?
Like physicians, anesthesia, or all other specialties?
No, we are not by all means. In fact, we cannot say behind this specialty in terms of preparing. We are now working on enhancing the skills and knowledge of principal investigators. In fact, we have just last week, in fact, talked about campaign to recruit principal investigators. As you all know, the requirement for phase 2, phase 3 clinical trials to require a physician to be the principal investigators, and as also a rule by the GCP guidelines. But in terms of behind, no, we are not. In fact, we are probably more capable, and we are ready to be part of clinical trial at any time with our experience, with our capabilities. But principal investigators has to have maybe double efforts in order to be capable of conducting clinical trials in terms of responsibilities, in terms of knowing the logistics and the policy and the procedure. They have to be certified as principal investigators for conducting clinical trial, having the GCP certificate.
Thank you, Doctor. Thank you so much.
Any additional question?
Is there any certain certification per name that the pharmacist should take in order to be certified to conduct the clinical trials?
This is extremely important question. Thank you very much, Dr. Ama, for this. There is not a specific certification. However, there is like a certification for clinical trial team. So if you go to the ACRP, which is the Association of Clinical Research Professionals, ACRP, they have certain certification for any member of the clinical trial team, including pharmacists. But there is not specific, there is no specific certification to certified pharmacists to be clinical certified in clinical trials. But the GCP certification is required. بعض institutions or بعض sites, they require the pharmacists to be certified in GCP. Others, they are, because if they are part of this as sub-investigator, they are required to be certified in GCP. But again, there is general certification, but not specific certification for pharmacists to be responsible for clinical, part of a clinical trial.
Excellent. Doctor, we have one question from the audience. The question is, in alignment with other entities involved in clinical trial used to be a big concern with MNC. What do you think about that?
What do you mean by MNC?
Well, I don't know. Unfortunately, I don't know what is MNC means.
Can you repeat the question? Maybe we can figure out what
is. In alignment with other entities involved in clinical trial used to be a big concern with MNC. What do you think about that?
I'm sorry, the question is not clear to me without knowing MNC.
Yeah, I'm sorry for that.
I cannot answer the question. Maybe if they can follow up.
Maybe the requester. It's a multinational company. MNC means multinational company.
Oh, sorry.
So I'll repeat the question again. In alignment with other entities involved in clinical trial, used to be big concern with multinational company. What do you think about that?
I don't think this is true, because the multinational companies, it is maybe a concern for the marketing of products, but it's not a concern for conducting clinical trials, because usually the clinical trials, the sponsor is only one. It is not like different countries, different companies, but it can be from one company, from a country. The regulations and the guidelines for that particular company from their country, let's say Europe, it is not very different from North America. And again, it's not very different from Japan or from Australia. So we are here, we have the backbone of the policy, and that we are following, and everybody is following, by the way, is the ICH GCP. So everybody is going by the ICH GCP. It's international, international committee on harmonization. It's not individual companies, individual countries, I mean. So I don't think it is a concern for us or for other countries, if I understood the question correctly.
Excellent. Thank you very much, Dr. Majid Al-Jraisi, for this very excellent talk that we really needed at that time. We appreciate your participation in our event, and would like to see you soon in the next event. Thank you very much, Dr. Al-Jraisi.
شكرا للجميع. شكرا جزيلا.
Now we will move to the official opening ceremony with a talk from His Excellency Chief Medical Officer, Dr. Saad Al-Mahraj.
بسم الله والحمد لله والصلاة والسلام على أشرف خلق الله سيدنا محمد وعلى آله وصحبه ومن والاه. The Ministry of National Guard Health Affairs aim to provide the optimum health care to its patient safety as its core value and to provide the holistic and optimum therapy for patients. The pharmaceutical care services played a significant role in ensuring patient and medication safety as part of the multidisciplinary team and management of patients. To generate new knowledge for improving the therapeutic use of medicine and overall healthcare outcome, King Abdulaziz Medical City in Riyadh not only strive to provide the high quality of pharmaceutical services, but also actively engaged in pharmaceutical research and development. As we begin the year with optimism, despite the current pandemic, pharmaceutical care services continue to explore ways to improve patient outcome through research. Thus, the launching of the first virtual pharmacy research to be held virtually on January 6 and 7, 2021. And as part of this significant event, I have the pleasure to introduce the book of abstract that reflect the hard work and commitment of our colleagues in the pharmaceutical care services. Our heartfelt appreciation goes to the custodian of the two holy mosques, King Salman bin Abdul Aziz, and to His Royal Highness, the Crown Prince Mohammed bin Salman bin Abdul Aziz, for their unwavering support to advance the healthcare services within the kingdom. And Prince Abdullah bin Bandar bin Abdul Aziz, Minister of National Guard, for his consistent patronage for all healthcare activities. I would also like to thank His Excellency Dr. Bandar Al-Ginawi, Chief Executive Officer, for his engagement and leading the transformation project in line with the Health Affairs Core Principle for providing the best quality and safe patient care. May I also take this opportunity to acknowledge the great effort exerted by Dr. Maha Al-Ammari for leading this elite group of pharmacists, as well as to the committee member for their hard work and dedication to bring this activity into reality through the able leadership of both Dr. Khalil Al-Majid, Acting Executive Director Operation, and Dr. Saad Al-Ubaidi, Acting Director Pharmaceutical Care Services. Once again, my utmost gratitude to each and every one of you who participated in this event. We promise our continuous support to medical and pharmaceutical research, as well as our commitment to maintain the highest level of standard in healthcare services. Wassalamu alaikum wa rahmatullahi wa barakatuh.
Bismillah ar-Rahman ar-Rahim. It's a time now to our scientific program to be launched. We'll be waiting for all the judges for the first morning session to join us on the Microsoft Teams. Most of them are around, but we are still waiting for one of them. So bear with us. We'll get back soon to you. السلام عليكم.
وعليك السلام.
من فضلكم اللي فاتح المايك ممكن يسكروا من طرفه لأنه في عندنا أصوات هنا. We are still waiting for the whole judges and the whole morning session speakers to join us, and we are arranging this. So بس كده أعطونا 5 to 10 minutes and we will start إن شاء الله. السلام عليكم. دكتور أوس الشمسان موجود معنا؟
موجود دكتورة.
حياك الله بروف. دكتور ثامر الشمري؟ دكتور عبد الله الثميري؟
السلام موجود دكتور.
حياك الله دكتور.
يك.
دكتورة هند مدميغ؟
هلا دكتورة، أهلا معك.
حياك الله دكتورة هند. So ننتظر دكتور ثامر الشمري to join us and we'll start the session.
على خير إن شاء الله.
بسم الله الرحمن الرحيم. راح أرجع أتأكد من أسماء المحكمين معانا. دكتور أوس.
موجود دكتورة.
دكتور عبد الله الثميري.
موجود دكتور.
دكتور ثامر الشمري.
حياك الله دكتورة.
دكتورة هند مدامية. دكتورة هند مدامية.
أنا موجودة، سمعتيني دكتورة؟
أيوة شكرا لكم. So we'll start our session today by introducing our judges. Dr. Aus Shamsan is a professor and dean of the College of Pharmacy at KSAU University since 2017. He held the position of a director of research center at the College of Pharmacy at KSAU University for 1 year and a co-director of the Joint Center of Excellence in Nanomedicine at KCAST. He's a director of King Abdullah Institute for the Nanotechnology for 3 years, and he earned his PhD degree in pharmaceutical science with a specialization in nanobiotechnology from the University of Alberta, Canada in 2010. He worked in the development of cancer vaccine in NSCLC and worked as a postdoctoral fellow at the National Institute for Nanotechnology, National Research Council in Canada. Dr. Shamsan received several international awards for his researches and achievements in nanotechnology, and currently he is the chair of the Future of Health Task Force and S20 Engagement Group. And he is the editor-in-chief for the Saudi Pharmaceutical Journal. So welcome, Dr. Shamsan. Dr. Thamir Shamari received his PhD degree in pharmacoepidemiology and pharmacoeconomics, and he's a well-known figure in the Kingdom of Saudi Arabia in the pharmacovigilance field. He received his degree from University of Rhode Island, USA. Currently, he's an associate professor of health outcomes at College of Medical Applied Science and College of Pharmacy at Riyadh Al Ilm University. Dr. Shamari is a pharmacoepidemiology and pharmacovigilance consultant and served as a director of the National Pharmacovigilance and Drug Safety Center at the Saudi FDA. His area of interest and research include patient and medication safety, pharmacoepidemiology, and pharmacovigilance. Dr. Shamari is very active in research, and he has over 80 publications in a peer-reviewed journals. So welcome, Dr. Thamir. Dr. Abdullah Thmeri is an assistant professor at the Department of Clinical Pharmacy, College of Pharmacy at Prince Sattam bin Abdulaziz University, Al Kharj. He earned his bachelor degree in pharmaceutical science from King Saud University in 2008, and he earned his PhD degree in pharmacoeconomic and outcome research from Nova Southeastern University from the States in 2016. Dr. Thmeri has been an active member in ESPER since 2018 till present with a multiple presentation and publications. So welcome, Dr. Thmeri. Dr. Hind Mdamir is a very well-known figure in cardiology pharmacotherapy. She's a clinical pharmacist specialist in cardiac science at King Abdulaziz Medical City in Riyadh. She is the Specialized Cardiology Residency Program Director and the Associate Professor of Pharmacy Practice at College of Pharmacy, King Saud bin Abdulaziz University for Health Science. Dr. Mdamir had led many researches and participated in others as a co-author. Her researches were published in peer-reviewed journals as well. And currently she's holding the position of Saudi Medication Safety Chairman at Ministry of National Guard Health Affairs with a focus on quality initiatives and to improve patient safety and medication safety as well. So welcome our judges. And we'll start our first session this morning with Dr. Lulwa Sweden by presenting a topic of mental health outcomes amongst healthcare workers during COVID-19 pandemic in Saudi Arabia. Dr. Lulwa Sweden, you can start sharing your slides, please, and you have 15 minutes, including question and answers from the judges.
Thank you, Dr. Maha. Assalamu alaikum. Did you hear me and see the slides?
Yes, go ahead, please.
Thank you, Dr. Maha, and the organizing and scientific committee. It's a great pleasure to be with you today and to participate in the first Pharmacy Research Day. I'll take you back in time in March 11, 2020, when COVID-19 was declared a pandemic by WHO. After that date, almost everyone was affected emotionally and physically from the dramatic change in our daily living. And looking at healthcare providers, we see that they are mostly susceptible to emotional distress due to their exposure to the virus when treating the affected patients and the decisions they are faced to make on a daily basis. Also taking into consideration their loved ones, and the list goes on. So in the context of COVID-19 pandemic, it is essential to evaluate the mental health of healthcare workers, hence came the idea of the research I'll be presenting today. So the aim of the study was to assess the mental health outcomes and associated risk factors among healthcare workers during COVID-19 in Saudi Arabia. The secondary objectives were to evaluate the severity of symptoms of depression and anxiety and insomnia using validated scales, and to evaluate the associations between the severity of symptoms and different categorical variables. To achieve that, we worked on a cross-sectional web-based survey administered using Lime software, and the intended sample were healthcare workers from tertiary care and Ministry of Health centers across the regions of Saudi Arabia. The web link of the questionnaire was active from April 27 to May 4 of 2020, and it was sent to prospective participants through WhatsApp, Twitter, and official emails. The survey contained demographic and job-related questions, and the mental health of the participants was evaluated using the 9-item healthcare questionnaire PHQ-9 for depression, the 7-item generalized anxiety disorder scale, the GAD-7 for anxiety, and the 7-item insomnia severity index for insomnia. We had 1,130 participants in the survey, of which 720 completed it and were included in the final analysis. Of the 720 participants, female were double the male, counting for around 64% of the respondents. For nationality, Saudi were slightly higher than non-Saudi, and they were 57 versus 43% respectively. For the age group, the majority of the sample, around 75%, were more than 30 years of age. For the marital status, around 62.5% were single, 58.3% had children, and around 61% were living with their family. 35.6% were working from home, and 25.6% were isolated in a separate room. 84% of the respondents were from the central region. 76% of them were working in National Guard. For the departments, the participants belonged to 21.67% were working under critical care, and for pharmacy, they were 22.64%, and the majority of the respondents, counting for 35.3%, were from other departments, for example, physiotherapy and respiratory therapy. Nurses were the highest respondent, accounting for 36.4%, then physician 26.94%, then pharmacists 23.75%. 92% of the participants had postgraduate training and education, and around 58% of them had more than 10 years of professional experience. Around 62% of the participants had no previous experience with pandemic. Only 32.5% had some training for pandemic, and only 27.78% of the respondents were frontline healthcare workers dealing with suspected and confirmed COVID cases. So we assessed the severity of symptoms using the depression, anxiety, and insomnia instrument, and we reported the result in median and interquintile range. For the PHQ result for depression, which usually ranges from 0 to 27, the median score was 4, and the range was from 2 to 9. The majority of respondents, 50.8%, had normal symptoms, while 26.1% had mild symptoms. The remaining of the respondents, which count for 23%, had moderate, severe, and severe symptoms in total. For an example,
دكتورة لولوى. دكتورة لولوى. دكتورة لولوى.
I think she is muted.
She's muted.
Sorry, can you hear me now?
Yes, Lulwa, go ahead.
Okay, so for the insomnia symptoms, the median score was 6. 57% scored or experienced absence of insomnia symptoms, while 28.75% had subthreshold symptoms, and the remaining, which were around 14%, had moderate severe to severe symptoms of insomnia. So based on that, we used the chi-square to look for association between the demographic variables and instrument score. We find that depression and anxiety, insomnia were significantly associated with age, years of professional experience, and working position. Depression and anxiety were significantly associated with nationality and living with family. Depression only was significantly associated with marital status, having children, and working in Ministry of National Guard. For anxiety, it was only anxiety only was associated with the professional title. So based on that, we did multivariable logistic regression to find predictive risk factors for developing depression, insomnia, and anxiety for the following variables, and found the following. For the associated risk factor for depression, we found that being a non-Saudi and a second-line healthcare worker has a lower risk of developing depression, while living with your family isolated in a room had a higher risk of developing depression. For the anxiety, being a female was associated with a higher risk of was a risk factor of developing anxiety, and being a nurse and a second-line healthcare worker was a lower risk of developing anxiety. For insomnia, being a second-line healthcare worker was associated with a lower risk of developing insomnia. So in conclusion, the symptoms of depression, anxiety, and insomnia were reported in a lower proportion of healthcare workers in our study. The participants who were female frontline workers, Saudi, living with their family, and working from home in isolated rooms were predisposed to developing psychological disorders. Although the majority of the sample were a second-line worker and from the central region, and it's difficult to generalize, but it's an initial report, and we can work on future results and research from there. Thank you for listening, and I'm happy to have any questions.
Okay, I assume maybe we can ask a couple of questions. Dr. Thamer, you can start if you want.
Go ahead, Dr. Ross.
Well, okay, thank you, Lola, for the nice presentation. I like the work, but I have one question. What is the logic of choosing this regression model, the multivariate logistic regression?
Because we were working with categorical data, and we wanted to find an association between the mental health and those variables. That's why we use the multivariable logistic regression.
So, do you think you have continuous data to use this model?
We had the score, but it's continuous data to use.
Yes, so whether your variables are continuous or discrete to use this model, do you think you have here a continuous data to use such a model for regression? Do you think you will get the right association with the multivariate logistic?
In my understanding that we can use this type of analysis in categorical data, and for the continuous data we use the logistic regression. I'm not sure how to answer this question.
Well, here you have scoring, so this is basically, yes, it's continuous, but on the other hand, you have variables that by nature are not continuous. So this type of regression would show you like a stimuli response type of curve. So if you have one variable on one hand that does not go on a scale with increase or decrease, you cannot use this model for. So probably not all variables in your study can be applied for this model. So this is my comment. Dr. Thamer.
Thank you, Dr. Lula. Very nice work. Actually, I have a couple of questions. Since we talk about, Dr. Ross talk about modeling, I'm not sure if the model you have used it's really accurate for different reasons, and the results from that. And I would like to ask you, Lula, do you believe in the results that you get, the association between the mental health and the COVID-19, especially when you talk about the nature of the study and ignoring different like confounders or different variables you are missing? Here, talk about disease, we talk about medication, we talk about different like co-founder. So do you think this is an accurate measurement or to do based on the nature of your study?
You're right, Victor. There are some limitations for the study that we did not take the history of depression, any available comorbidities that are on the social support system, and the workload was not taken into consideration in the study and will have an impact on the end result.
Yeah, go ahead.
So as I mentioned, we cannot generalize the result of the study because of some of the limitation available. However, it gives us an idea how was the result of the mental health assessment during this time, taking into consideration the limitations.
Okay, generalizability is different. What we talk about it, Lula. This is different because your most of your population is the central region, so it's a little bit different. Let's move to other question just for sake of time. You mentioned that you have you did a web-based survey and you sent it by WhatsApp and etc. So was this open to everyone or for selective population or selective people?
It was sent to healthcare providers.
So do you know how much got this message?
We know the number of people who entered the link, which were 1,130.
Okay.
And the ones who completed the survey.
But this, Lulwa, do you know if someone sent it to other person?
No.
Okay, so this is difficult with the web-based survey to know how people access this survey, how people complete the survey. So your response rate, despite it's a little bit like 64, which is a little bit okay, but this is one of the limitations for web surveys. So we don't know how many people like see the survey and apply. So keep this in your mind. Well done. Thank you.
شكرا لكم.
Thank you, Dr. Aus. Thank you, Dr. Thamer, for your valuable questions and comments. Shall we move now to the second speaker? Yeah, Dr. Aus, Dr. Samer?
Yes,
ready?
please.
Yes.
So our second speaker today is Dr. Mubarak Al-Dosari, and he will present development of mRNA-based vaccine candidate against COVID-19. Dr. Mubarak?
Assalamu alaikum.
Wa alaikum salam wa rahmatullah. Can you share your slides, please?
Yes, just a few minutes. Can you see it now? Hello?
طالع. لا يا مبارك, we cannot see your slides.
And now?
Yes, go ahead and share your slides. Tawfiq.
Thank you, Dr. Maha. My name is Mubarak Al-Dosari. I am pharmacist at National Guard Health Affairs and postgraduate student at KSAU University College of Pharmacy, Master of Toxicology degree candidate. Today, my subject is about development of mRNA vaccine candidate against COVID-19. First of all, let me explain how the SARS virus infects the human cell. First of all, here is in the green color is the spike protein, which will interact with the angiotensin converting enzyme receptor, which allow the coronavirus to enter the human cell and infect the human cell through the release the genome and translate it in the ribosome. After that, the coronavirus will use the human cell as biomanufacture to replicate itself and synthesize more and more of coronavirus and infect another cells. It's used to know that the mRNA is fragile and unstable. Starting from 1960, there is significant advancement allow us to use it as a vaccine. Today. Especially in 1990, Dr. Wolf demonstrated that mRNA injected in mice can be translated into protein. There is several obstacles that prevent us from using it as a vaccine from a decade ago. RNA is designed to be unstable. RNAs are everywhere. Triggers strong innate immune response. Also, no enzyme available to make good manufacture practice material. The sequence data available was limited. But today we have advancement in the technology allow us to use the mRNA as a vaccine. Like here in the first, capping of 5' and polyadenylation at the 3' will enhance the stability and will increase the translation efficiency. Codon utilization will facilitate the ribosome binding. Also, modification of the nucleotide will control the immune response and also will especially the innate immune system and increase translation yield. Inclusion of self-amplification will reduce dose and cost. I will speak moreover later about it. Improved delivery mechanism will protect RNA from degradation and will increase the uptake by the cell. In comparison with other platforms, mRNA vaccine has several advantages like non-infectious and rapid development, which we need. For example, in case of corona epidemic now, also it's degradable. That means there is no risk or less risk for DNA integration. Now, let me talk about the overview of the mRNA technology. First, the antigen will be cloned in the specific plasmid. That plasmid will be transformed in the E. coli for amplification. After that, plasmid will be isolated and linearized by restrictions enzyme. Now we can use it as a template for in vitro transcription to synthesize our mRNA. After that, the capping stage, we will cap our mRNA with in the 5' region to increase the stability of our mRNA. In the purification stage, actually, we will check for the integrity and the purity of our mRNA by HPLC and gel electrophoresis. Here is our, there is two mRNA types. First, mRNA construct and the self-amplifying mRNA construct. I will speak first about the mRNA construct. Here is the gene of interest to be in the middle, and we will utilize the surrounding genes to increase the stability, like here in the 5' and also in the polyadenylation here, also to increase the efficiency of the translation. Also here, untranslated region, also to we found it's reported that addition of this untranslated region will increase the mRNA half-life. Let me compare it with the self-amplifying mRNA construct. Here is non-structural gene for self-amplification. It's upstream the gene of interest. This will allow the mRNA to replicate itself, as it shows here in the figure down. You can see it. There is one mRNA, it's replicated itself and yield more of antigen after translation. Here is the original or the ordinary mRNA. Here is one mRNA will be translated and given an antigen. So there is challenge for this mRNA type, which is longer sequence, and it will be more vulnerable to error. Also, strong response and may limit the antigen production. Prolonged replication mRNA decrease the viability of the host cell. Potential risk for anti-factor immunity. Once the mRNA enter the cell, will be entering the phagosome, and it will release the mRNA. Here is our gene. First, it will stimulate the toll-like receptor, and it will activate the interferon type 1. Our mRNA will be translated through the ribosome and release an antigen. Our antigen will be represented on the MHC1 and MHC2 for activation of the immune system. And here is the, let me talk about the workflow of our project. Here is the viral isolation. We are focusing on the viral isolation to isolate the virus from the patient within Saudi Arabia. For the sequencing, we are sequencing with collaboration with KMark from this samples from the patient with Saudi Arabia, also from another centers in Saudi Arabia for the patient in Saudi Arabia. And with mRNA technology, as I said before, we will design our antigen and clone it in the plasmid, then amplify it in the E. coli, and then with in vitro synthesis, we will synthesize our mRNA. Here is for the quality control. Now mRNA analysis and mRNA purification will be checked for the mRNA integrity and purity. In vitro testing and animal testing for the immunogenicity of our immunogenicity for our mRNA. As I said before, we collaborate with the Department of Infectious Disease at KMark with Dr. Majid Al-Ghuraibi. The most abundant sequence was, there's two sequences that are abundant in Saudi Arabia. Sequence 1383, which is 61.4 worldwide and 75% in KSA. And there is sequence 399, 16.6 worldwide and 25.8 in KSA. And based of that, we based on those two sequence, we designed our candidate to be 6 mRNA vaccine candidate. Here is we use the self-replicating and the also the full spike mRNA protein for the 1383, also self-replicating receptor for mRNA. Also, here is sequence 399. We use the full spike protein for that. We have 4 candidates of sequence 1383 and sequence 2 candidate sequence of sequence 399. Two vaccine candidates. Sorry. Thanks. I want a special thanks to Dr. Mish'al, my advisor from KSU, and also in Kmart, Dr. Majid Al-Ghrabi. Special thanks to him also, and special thanks to our sponsors, Ministry of Education and Kmart and King Saud University, and also for international collaborators. Back to you, Dr. Amha. Thank you.
دكتور مبارك.
هلا والله.
حياك الله.
يا هلا وسهلا.
Dr. Thamer, you can go ahead.
طيب. Thank you, Dr. Mbarak. Very nice work. Actually, I have some questions. First, I was following up with you during a presentation, but you stopped there. I was expecting to get over more results from your talk. So I just want to ask a question because I have another question for this. When did you start this project, Mbarak?
We started about maybe in August. We start to build the base for this project.
Since August, so what was the purpose of this project, Mbarak? So to develop a vaccine, right?
Yeah.
So why you think our strain or the mRNA technology will be different than other technologies that we're doing outside or different places? I'm just trying to understand the exact purpose of the project, because I'm not following up with you with this project.
Yeah, the exact purpose of this project is to synthesize or manufacture mRNA vaccine for COVID-19.
Yes, go ahead.
Yeah, especially for sequenced patient, COVID-19 patient from Saudi Arabia.
Saudi Arabia.
And we will. That's also we want to see if it's more efficacy more than the others vaccines.
So do you mean that the mRNA technology will be different by different sequences among different population?
Yeah, that's what we were trying to investigate.
So and you did investigate, right?
We are still now under development, and there is no result yet. But inshallah, we will get it. Inshallah.
Because I think in one slide they said like 60% of the virus were similar. We have 75%. We're similar to 60% worldwide, right?
Yeah, that's it. Yeah.
So it's almost same.
Yeah, that's it. Yes, that's almost the same.
Okay.
Yeah, but also for this project, you mean our goals in this project? Also, we want to like training that's including me, of course, as a student. For if we have a next epidemic in the mRNA, will be trained to synthesize a vaccine for mRNA at the national level.
Okay, so for this for the COVID, will not continue to develop a vaccine from your side.
Sorry, again.
For this project, there's no plan to develop a vaccine.
We will try, and if we could, we will of course proceed with it.
Okay, shukran. Thank you, Dr. Mbarak. Dr. Aus.
Thank you.
Thank you, Barak, for the presentation. And here I also echo what Dr. Thamer mentioned. Actually, the presentation stopped bluntly, and we didn't see much of a result. So, given that you started in August, 4 months ago, I understand the pandemic situation affected all wet lab research, so probably this what puts your work behind a little bit. But I have to comment also on the logic. In vaccine development, usually starting from clinical isolates would start where the epidemic happened originally, where the outbreak happened. And this, for this specific virus, the gene sequence was published in January. So on January 11, the full genome of the COVID-19 virus was published. And from that, all companies were starting to develop vaccine accordingly without going into isolating clinical isolates from patients. But I understand the other point that you mentioned, that this is a good training to localize the know-how here, but it might not help the purpose of this particular vaccine. So it seems to me that you are reinventing the wheel in this project. But anyway, I would ask you about your future work. Well, now after you isolated and sequenced the virus, and you recognized the most abundant sequence, what will be the next step in generating the vaccine? What will you do?
Sorry, I didn't get your question.
What is your next step in developing the vaccine?
As I said before, our next step will be manufacturing mRNA vaccine, and if we see if it's the efficacy is more than the other vaccines that designed outside Saudi Arabia, we will proceed and we will manufacture it, of course.
Okay, let me rephrase my question. What test you will do to measure the efficacy of the vaccine?
Yeah, as I said before, we will have naive animal, and we will test the immunogenicity of it, and also there is in vitro test for macrophage and for the dendritic cell to test the immunogenicity of it.
Okay, thank you, Mbarak.
Thank you.
Thank you, Dr. Aus. Thank you, Dr. Thamer. Thank you, Dr. Mbarak, for your presentation. Shall we proceed now to the next speaker? Dr. Jihad Al-Ghamdi, are you around?
Assalamu alaikum.
Walaikum salam, Dr. Jihad. Dr. Jihad will present a topic on targeted next generation sequencing of genes involved in warfarin pharmacodynamic and pharmacokinetic pathways using the Saudi warfarin pharmacogenetic study SWAP. Tfadal, Dr. Jihad.
Assalamu alaikum, sabah al-khair. Just to make sure my slide is there.
Yes, it's clear. Can you share it, please? Thank you.
Yes. Good morning, everyone. And at the beginning, I would like to thank the scientific and organizing committee for organizing such a great event for research, pharmacy research. So today I'm going to present our study on doing targeted next generation sequencing of gene involved in warfarin pharmacodynamic and pharmacokinetic pathway. I will start my talk with a brief background about the warfarin pharmacogenomics and then giving details about the methodology in our study using describing the study cohort and the genetic analysis that was done in this study, and then completed my talk with providing the result, mainly the patient characteristics and the genetic variant that we found to be associated with warfarin, strength and limitation of the study. So as a background, we know that warfarin is an oral anticoagulant that is widely used for treatment and prophylaxis against thromboembolic events. However, the dosing of warfarin is proven to be complex and challenging in clinical practice for several reasons. Mainly, the warfarin has a narrow therapeutic index window, which means that it's required frequent monitoring and dose adjustment for the patient to be within the therapeutic range. It has also a nonlinear dose response, which means that a small change in the dose may result in a significant change in the response. And the most importantly, that the there is a patient variability in terms of the dose requirement. And that variability is proven to be due to several factors, including the genetic variant that is the focus of this study. However, also other factors such as the diet or drug-drug interactions play a major role. For the genetic factor, there are a lot of guidelines has been proposed by several entities, mainly the Clinical Pharmacogenetic Implementation Consortium, the CPIC, published a guideline for pharmacogenetic or genotype guided warfarin dosing algorithm. So mainly, and according to also the FDA and other organizations, they don't require to genotype the patient before prescribing the warfarin. However, if the genotype information is available for the patient, then that can be taken in consideration while dosing for the warfarin. And mainly, most of the guidelines focus on two genes, the vitamin KRC1 gene and the CYP2C9 gene. So if we know the genotype of these two enzyme, mainly one variant in each gene, then we could we could tailor the patient dose based on the genotype. And here is one algorithm, genotype guided algorithm that's been proposed by IWBC, which take into account several factors such as the age, height, weight, and the race, and the enzyme inducer, and taking enzyme inducer and inhibitors. This is from the part of non-genetic factor, and then also take into account the vitamin KRC1 genotype and CYP2C9 genotype as one of the genetic components. So in total, when you do this for your patient, you could end up with a value for the weekly dose that could be tailored more specifically for your patient. However, we know that these were designed based on European population mainly, or other populations not specifically focused and not necessarily to be true, holding true when it comes to our population. And that's what we need to do to find out if these variants are significant in our population in order to develop a more targeted, specific genotype guided algorithm for our population. So we aim in this study to perform a genomic profiling of gene involved in warfarin BK and BD pathway, mainly to identify common and rare, including novel genetic variant associated with warfarin responsiveness in the Saudi population. And then in the first part, we want to estimate the genetic determinant of variability in warfarin dose, meaning that we want to know how much of the warfarin dose variability can be attributed to genetic factor in the Saudi population. This can be seen as the first step or the first part of this study, and the second part we will do more targeted genotype guided algorithm. Now, we started this study. It's a prospective multi-center cohort study conducted here in KMC mainly. We included Saudi patients, and that's very important because we wanted to be very specific in terms of the nationality, in terms of the population, in terms of the genetic background. And we only included above 18 years old patients and those who are prescribed warfarin. We excluded patients if they have a prolonged baseline anticoagulation profile, or if they have a clinical indication for warfarin that required INR above 3, mainly for indications such as mechanical heart failure, if they are receiving chemotherapy, or they have confirmed diagnosis of chronic viral disease such as HIV or Hep A, B, C. Now, and this study was approved by the IRB of King Mark all patients were consented before collecting the blood. We followed up the patient for 10 days. This follow-up was conducted either as inpatient or outpatient in the anticoagulation clinics, and we designed a targeted multiplex next-generation sequencing panel that include the coding sequence, UTR, flanking region of 16 selected genes. These genes were mainly implicated in warfarin BDBK pathways. Now, for the aim of this study, we used the weekly dose as the outcome of this study. We calculated the weekly dose as the sum of warfarin dose from day 4 to day 10. And then we conducted a single genetic variant for studying the association between all the identified variant in our population against the warfarin dose. We used a linear micronization model, adjusting for age and weight and warfarin history status. And then for the top significant variant, we taken this further into stepwise multiple regression model to compare between a model that includes genetic components and a model that only based in non-genetic components. Now, coming to the result, we included 936 patients, a total almost 1,000 patients. 50% of them were female and 8% were smoker. Most of the patients, almost half of them, were received warfarin because as AFib. 18% for DVT and another 18% for BE. With regard to the comorbidity, 50% of our patients were diabetic, 70% of them were hypertensive, and 42 of them were hyperlipidemics. Now, coming to the other characteristics, most of the patients were elders with the mean age 62 years old. Almost also they are slightly obese with the average of BMI as 30. They received on average 3.6 milligram of warfarin dose on weekly. Only if we calculate it as a weekly, it was 25 milligram, and most of them they required on average 6 days to reach the stable INR. Now, these are the genes that were designed. These are the genes that we sequence and designs, and as you can see, most of them has some rules and either an pharmacokinetic pathway of warfarin or pharmacodynamic pathways, and these are the number of the identified variant within each gene. And as you can see here, the VKRC was one of the least genes with regard to the identified variant. Almost 11 variants were identified within this gene. However, most of the variants were exonic in the exonic area, which means that they were on the coding sequence. Now, coming to the significant variant, this table shows the top significant variant that were identified to be associated with warfarin dose, and as expected, we have identified this variant. The top one was already published in other population, has a very robust evidence to be associated with warfarin dose, as you can see with the B-value minus 49, and the A1 here showing the minor allele. In our population, we found that having the C allele is on average associated with a higher required dose of warfarin. Again, as expected, we identified this variant in genes that are already published. However, we have identified a more specific variant within the gene, and if we can compare our finding with, for example, the CBIC guidelines, we can see that the one implicated, the one reported in the CBIC, CYP2C9 star 3 and star 2 were not the top significant variant in our populations. And if we were using the same guideline in our population, we might not get the best possible result, as we found other variant within our population, such as this variant in the 2C9, which is more significantly associated with warfarin dose. So that's showing that there is a population differences, specific factors, the genetics variant that already reported in other population, not necessarily to be the same in our population, and developing genotype guided algorithms based on the result reported elsewhere may not be the best option in our population. Now, We have taken the top three variant and into the next models, and we have developed 8 models based on the stepwise multiple regression models, including significant non-genetic factor and significant genetic factor, to see how much of the warfarin dose could be explained by these variables. And as you can see here, if you look at the first 5 models that were not including any genetic factors, we could see that these variables, warfarin status, age, weight, indication, if the patient were indicated for AFib, having no other comorbidity, no drug-drug interaction, will explain around 25% of the variability in the dose on the warfarin weekly dose. However, if we just included one extra genetic variant, then the explained variability would increase up to 40%. And if we included the 4-3 variant, then the variability that could be explained is 43%, which is almost near to 50%. And that's showing that including accounting for the genetic factor is very important when it's come to the warfarin dosing. Now, aside from this, also we have looked to other genetic variant in actionable pharmacogenetic gene that were also reported in either pharmacogenomic database or CPIC with A1 level evidence to be associated with the drug response. And we have found some other genetic variant, other SNPs, and that could be also very important for dosing of clopidogrel or amitriptyline, citalopram, or other variants such as atorvastatin. So our main finding, we have found 165 novel variants and replicated 545 variants that already reported in elsewhere. Most of the variants, as expected, 89% of them, they were rare as expected, alongside also with other findings from other literatures. 8 variants within vitamin K or C, CYP2C9, CYP2C19, were significantly associated with warfarin dose, and the addition of the top genetic variant within this gene almost doubled the explained variance from 25 to 43%. Now, the strength of our study that we have utilized a relatively very large sample size, almost 1,000. And if we compare this as one and to other pharmacogenomic studies, it tend to be very large. Usually, the pharmacogenomic studies usually tend to be small. It's not like the other because of it's very difficult and challenging to follow up and recruit patients and collect the drug response as a phenotype. We have also utilized an NGS approach, and if we have relied on genotyping approach, we would miss around 23% of our findings that are very specific, novel to our populations. And then we have also used only Saudi population study, which provide a very comprehensive insight into the genetic variability in gene related to warfarin responsiveness in the Saudi populations. So this would be very specific to our populations. In terms of the limitation of our study, again, we have used a targeted panel. So we are really focused on 16 genes, and we have missed other genes that were not within our panels. And we were limited in terms of follow-up for the patient for only 10 days. And the next phase, we want to follow up the patient, giving that electronic health record would allow us to follow up the patient for a longer time period. At the end, I would like to thank the research group, mainly Dr. Mahal Ammari, the PI of this study, and Dr. Badam Zani, Dr. Mohammed Abloui, and Ms. Khazra Sultana. Special thanks to Medical Genomic Research Department and Saudi Biobank from KMARC and the Pharmaceutical Care Services from King Abdulaziz Medical City. With that, I conclude my talk. Thank you.
Thank you very much, Dr. Jihad.
Thank you.
Dr. Thamer, if you allow me, I have just a very quick question to ask Dr. Jihad. MashaAllah, the work is really amazing and the presentation is great.
Thank you so much.
I like the finding. I like focusing on our population. This will help in implementing precision medicine in the country, and hopefully we would see more impact of such study and similar studies in changing the medical practice and pharmaceutical care, inshallah, in the kingdom. My question is, what would you think probably the confounders that you haven't accounted for in this study, and would your patients receiving the same brand of warfarin, would this be considered one of the confounders, yes or no?
Absolutely, Dr. House. That's the type of we don't know, making sure the pharmacoequivalence of the medications and different brand might not be similar. So that's absolutely could be one of the confounder, knowing exactly what brand of warfarin. We know that medication differs in terms of the potency, maybe in terms of the concentration. So that's an absolutely one of the confounder that could be taken into account. But my understanding is that all the patients receive the same brand, the same type of medication. So that may overcome this. That would be true if we have different type of or different brand of the medication.
Okay, thank you very much, Dr. Jihad.
Thank you.
Very nice work. Well done. يعطيك العافية. Dr. Jihad, just want to ask about the time that you conduct the study, and if you just give some light on the type of population that you have studied. I'm assuming it's within the National Guard, which is like close population. So, and this is, do you think this will be a representative of the whole Saudi population? So if you can comment on these 2 points.
Yes, I think it is mainly within the NGHA, and whether that could be a representative, I would say it would be a representative of the Saudi population, despite that we know that the population has specific features, meaning that mainly they are coming from a military background, but that's we have to take into account that we don't ascertain the patient based on whatever. So all the patients who are coming to the clinics and they are willing to participate were enrolled in this study. So that could, to some extent, reduce the impact of the bias. And we know that there is a huge population served by NGHA. I think it's almost 1 million or so. So that's also, again, will reduce the bias of the population-specific factors in terms of, for example, coming from backgrounds, the same job background or the same ethnicity. Most of these patients were Saudis, were certain for the nationality. They were not as certain for anything else beside that. So that will reduce the risk of bias, I would say, although it will still exist, but it will be reduced significantly.
So the other point, Dr. Jihad, during your conducting the study, I'm assuming that some of the patients have different type of diseases, different type of comorbidities among these patients. Do you think this would have a role in the variant of these genes or sequences you've done? Did you consider that?
Yeah. In the non-genetic model, we have entered all the comorbidities into the models. We have entered all the indication into the models, and then it was done as a stepwise model. So whatever factor that were not significant, it was rolled out by the model. So these factors were found as non-significant, I think, except for having no comorbidity. So if you have any kind of comorbidity, then that will be a count. If you have no comorbidity, I don't know if you can see the slide here. So having no comorbidity is a significant, but having any type of comorbidity would influence that, but that would not be fair to what exactly the type of comorbidity. And as you can see in the characteristics of our patient, most of them, I would say 50% were hypertensive, 70% were hypertensive, 50% were diabetics, and 40% were hyperlipidemic. So most of the patient actually with other comorbidities. However, the model excluded these. They were found non-significant in the model.
Thank you. That's the last question, Dr. Jihad. We know that warfarin is really important. It's really like a drug that we need, and we'll continue to have this drug. But now, like moving to NOAC as anticoagulants, the novel oral anticoagulants. Do you think it's like cost effective to do such like that? Was effective, but do you think it really will have a big impact going to use a lot of new oral anticoagulants?
Yeah, I can see. I think this is need further research to be able to answer this with certainty. However, as you could see, as you say, that warfarin will still be existed, and I think the cost of the genetic doing a genotyping is getting less and less with the time. So with the time, it might be cost effective to test your patient first if they were found to have a genetic variant that would increase the risk of warfarin risk events like bleeding or hemorrhage. Then you shift only those patients to the other agents, which will be more cost effective than starting your patient immediately to more expensive medication.
Thank you, Dr. Jihad. Well done.
Thank you so much. Allah يعطيك العافية. Thank you so much. بالتوفيق.
Thank you, Dr. Jihad. Thank you very much.
Thank you. الله يعطيك
Thank you,
العافية.
Dr. Aus. يعطيك العافية. Thank you, Dr. Aus. Dr. Thamer.
شكرا دكتورة. Thank you for
شكرا.
this session. Thanks.
نشوفك في السيشن اللي بعده دكتور أوس إن شاء الله. Our next speaker is Dr. Raed Kinsara. Dr. Raed, is he around?
Hello, السلام عليكم.
عليكم السلام, حياك الله, Dr. Raed. Dr. Raed will present his talk on the impact of implementation of computerized insulin order set for the control of hyperglycemia in hospitalized cardiac patients with type 2 diabetes mellitus at a tertiary hospital. تفضل Dr. Raed. Can you share your slides, please?
Yeah. Does it appear?
Yes, it appears. Can you display it?
That's good.
ممتاز. تفضل بالتوفيق إن شاء الله.
So good morning, everyone. First of all, I would like to thank the organizing and the scientific committee for conducting this special event, which I hope will continue in annual basis, and also for giving me this opportunity to presenting my research to you, which is about the impact of implementation of computerized insulin order set for the control of hyperglycemia in hospitalized cardiac patients with type 2 diabetes. Diabetic patients have a fourfold greater risk for development of cardiovascular diseases compared to patients without diabetes. And poor glycemic control in those populations can lead to several unwanted outcomes. For example, can lead to increase the risk of thrombosis, risk of infection, and also can increase the length of hospital stays and operational cost. So the Institute for Safe Medication Practices, the ISMP, has recommended to use a structural computerized insulin order set to improve the patient care. To date, there is a gap in the literature about the effectiveness of insulin order set for the control of blood glucose level in those populations in Saudi Arabia. So therefore, our study aim is to determine the effectiveness of computerized insulin order set in controlling blood glucose level in those patients. It's a quasi-experimental pre- and post-study design was conducted at King Faisal Cardiac Center at King Abdulaziz Medical City in Jeddah between June 2017 to August 2019. The study has received the IRB approval from the KMARC in September 2018. The primary endpoint of the study is to determine the mean differences in random blood glucose level before and after the implementation of computerized insulin order set. The secondary endpoint including mean differences in fasting blood glucose level. the proportion of patient achieving the target random blood glucose level, which is less than 180 milligram per deciliter, number of hyperglycemic episode, which is any blood glucose reading above 180 milligram per deciliter, and hypoglycemic episode, which is any blood glucose level less than 70 milligram per deciliter, and physician adherence to the order set. We included adult type 2 diabetes patients who were hospitalized for at least 3 days during the study period. We excluded type 1 diabetes patient undergoing a major cardiac surgery like CABG or a heart valvular replacement, patient on enteral or total parenteral nutrition, patient with active malignancy, or patient on regular hemodialysis. The study consisted of 3 phases. The pre-phase, which include the patient who met the eligibility criteria of the study and admitted to our cardiac center. And then we did a data collection that included baseline characteristic and blood glucose level, 3 random and 1 fasting, between June to December 2017. The second phase is the interventional phase. And in this phase, the insulin protocol was designed and reviewed by the endocrinology department, and then this protocol was approved by the pharmacy and hospital pharmacy and therapeutic committee, and then this protocol was integrated into our electronic system as an order set, and after that several educational session was conducted to the cardiology department, including physician, nurses, and clinical pharmacists about how to use this order set between May to December 2018. The third phase is the post phase after the integration of the order set. In this phase, it's similar like the pre-phase. We included the eligible admitted patient to our cardiac center between February to August 2019. This is our insulin protocol, and the protocol has divided the patient in 2 groups. First group is the patient with good oral intake, and in this group of patients, they should receive basal and bolus insulin regimen. The second group is the patient with poor oral intake, and in this group, the patient should receive basal plus correctional insulin regimen. So the use of a sliding scale insulin as a monotherapy is totally discouraged. This is the protocol after integrated into our electronic system. So we determined 97 patients in each phase, total of 194 patients, to provide a 90% power to detect a mean difference of 29 milligram per deciliter for random blood glucose level before and after the intervention. The study has 95 confidence interval with an alpha of 0.05. Descriptive statistic was used to present the baseline characteristic. We performed the student t-test and Manin-Whitney test to determine the differences between means and medians of the continuous variable, Chi or Fisher-Exact test for categorical variable, and also we performed a regression analysis to adjust for any potential confounders between the 2 phases. For the result, this is the baseline characteristic, and there was no statistical significant difference between the 2 phases, and the majority of the patients were male and obese, and also the majority of the patients were admitted as a case of acute decompensated heart failure and ischemic heart disease. In terms of diabetes home medication, also there was no difference between the 2 groups in both oral hypoglycemic agent or insulin. The majority of the patient also had hypertension as a comorbidity, and there was a difference between the 2 groups in terms of ischemic heart disease, atrial fibrillation, others like hypothyroidism or pneumonia. For the primary endpoint, the mean random blood glucose level was 201 in the pre-phase compared to 171 in the post-phase, with a p-value of 0.001. And this finding also has consisted with the mean reduction of blood glucose level of 32.4 after we performed a regression analysis, and the adjustment was based on age, gender, ANC, ischemic heart disease, deep vein thrombosis, and baseline serum creatinine. For the secondary endpoint, the median fasting blood glucose level was 166 in the pre-phase compared to 153 in the post-phase, with a significant p-value. Only 29% has achieved a random blood glucose level target in the pre-phase compared to 63% in the post-phase, with a significant p-value. The number of hyperglycemic episodes were 867 in the pre-phase and was reduced to 548 in the post-phase, with also significant p-value, and there was no significant difference between the two groups in terms of hypoglycemic episodes. The adherence to our order set was 70%. The study has several strengths. It's the first study to assess the impact of computerized insulin order set for controlling of blood glucose level in Saudi Arabia. It's a quasi-experimental study designed to assess the utility of the intervention. The study had 90% power with a sufficient number of patients, and also this insulin management protocol can be used for non-cardiac diabetic patients, especially in the medical ward and non-ICU setting. And also we were able to increase the physician awarenesses about and the importance also about the use of the structural insulin regimen in hospitalized diabetic patients. For the limitation, the adherence to the order set was only 70%, but even though we detect our target or difference of the study, the order set was not known nor accessible to some rotating medical resident initially, but after that we ensured the accessibility. Some patients didn't have 4 blood glucose reading per day, and it's a single center experience. So as a conclusion, the implementation of computerized insulin order set by a pharmacist was associated with significant improvement in random and fasting glycemic control without increasing the risk of hyperglycemia or hypoglycemia among hospitalized cardiac patients with type 2 diabetes. And by this slide, I end my presentation. Thank you.
Yeah, شكرا دكتور رائد. أنا هند مديمير. It's actually great idea, as a matter of fact. أنا حتى تحمست للproject. Apparently, it's actually a good project that should be considered for corporate incorporation. Look at the whole Ministry of National Guard, Health Affairs, or regional facilities. I do have a question. You're welcome. I do have actually a couple of questions, if you don't mind. If you go back to the the power of the study, do you know why did you actually reduce it? It's okay. It's 90%. But
yeah, 90%.
Yes. So would that affect your hypothesis or not? The power of your study, would it be affected if you lower it? And why would you lower it? Why didn't you go with the usual 95?
Because we did the calculation, and if we provide 80% power, the sample size will be decreased.
Okay.
The number of patients. So we want to take a lot of patients in this study, but also it was my residency research project, so it's we considered by the timeline. So I think if we take more power study, like 95, it's mean a lot. Also, the number of patients will be increased, and we were afraid that the time it will be out of our hand.
Okay. Okay. I see. Thank you. You mentioned also that the compliance rate with the protocol was about 70%. but you were able to detect it and correct it. How did you actually, you were able to detect it and correct it? How was that corrected? And how is that going to affect your results?
You mean because of the adherence to the order set was only 70%?
Yeah, 70%. It means 1/3 of your results are contaminated, correct?
Yeah, correct.
Yeah. So how is that? You mentioned that it was detected and corrected. How did you correct it?
It's not corrected. I mean detected. So we detect the adherence. You mean, or detect the difference?
The adherence. You said that the adherence. Yes.
Yes. Actually, because there was no comparing group, and we didn't set like a number to achieve about the order set, but we detect the adherence to order set if the physician use our order set for at least 3 days in total of 7 days. So we will consider this patient we will consider this step as adherence to the order set.
So does that mean, what did you do with the results if they're not adherent? Will you continue for the the post phase?
Yeah, we will. We include it in the post phase.
But don't you think, and you can correct me, but if that prescriber was not compliant with the protocol in the pre-phase, I mean, how could you actually generalize the results of the post phase then? Definitely the post phase results will be contaminated. Isn't that correct?
Yeah, correct.
But still, I believe you included the results, as you said.
Yeah, we include the result, and we put the adherence percentage to order set.
Okay. My last question, I do apologize. My last question, if you go to the hypoglycemia events, it was, I believe, 24 in the pre and then 33 in the post.
For the result, okay.
I know the p-value was 0.45, but
yeah, there was no difference.
Where in the hypoglycemia?
Yeah.
Hypoglycemic. So in the pre-phase, it will be it's actually lower. Okay. No, it's 24 and 33. I see a slight difference. I know statistically it's not significant, but still there is actually, which is weird. Why would patients have more hypoglycemic events? Sometimes we can go with the clinical, findings rather than just pure statistical significance. We go with them with the clinical significance, and I see that the number of events of hypoglycemic events, they were actually more. So that's my opinion.
Yeah, finally, because can I comment in your?
Yeah, of course, yes.
Because in the pre-phase, the physician were usually used a correctional or sliding scale insulin only, so the number of insulin doses were lower actually compared to the post-phase, because in the post-phase we use a structured insulin regimen that include basal and bolus insulin. So the number of insulin doses, of course, it's increased compared to the pre-phase.
Okay, thank you so much, Dr. Raed. Thank you. And I believe
Dr.
Mary.
Dr. Raed, yeah, I have a couple of questions. So first, I would like to congratulate you for the project. As Dr. Hind mentioned before, we need more projects like that where optimizing healthcare is needed. And with a resident like you, we can really optimize the healthcare. But I get the side of that you are on a time limit where you have to finish your project. But I again stress the sample size point that Dr. Hind mentioned, because it's really affected the whole project. So what I would recommend that going back and increase your sample size and actually have a methodology on how you actually follow up with the patients, and if you have contaminated sample size, as you mentioned, how you deal with that. It's very important to address this point for going forward with the project and improving the project. So you have really good project, but you can optimize it in that sense. Going back to related to the sample size, the confounding variables that you control in the regression, there is no clarification on the regression, but I would like to go back to the confounding factors that you control for.
You mean this part?
Yes. So this is a regression you control for the age, gender, and BMI. So from pre and post regression, you would think that it's the same sample size that you compare pre and post, but this is maybe the methodology that you actually use is different than the methodology you intended to use. So we usually control for other factors also, the clinical factors, maybe some different settings within the clinic has varied pre and post as well. So it's not just the clinical factors, also the system or healthcare system factors also important in this regard. Did you get what I'm saying?
We did actually we did a multivariant calculation depending on the regression analysis, and we chose different variables, and all of it was consistent with our finding. But we choose this because it's more it's contain more variables, and also it's contain some variables that there was a difference between the two groups in it, like ischemic heart disease or atrial fibrillation. That's why.
Yeah, but right, what I'm saying is it is the model, the regression model that used in this project can be optimized by using another regression model. So what I'm saying is you might go back and address this point and read on the pre and post analysis and regression and include different confounding variables, not just the clinical part, also the systematic part. So yeah, there is there's other confounding variables that might contribute to why the variation in glycemic control. And the last question is just not as a statistical question, but it's what do you think the result will be similar if you applied this in non-cardiac setting? Like you applied this in cardiac center, right? So do you think if you apply it, for example, in cancer center, would be the same?
It will be different in cancer center because the target, it will be different. But for medical wards in non-ICU setting, yes, can be the same because the dosing and the protocol, and also the target, it's similar between the two population. But for cancer, no, because the target blood glucose level, it's different.
Okay. Okay, thank you. That's all my questions and concerns.
Thank you دكتور هند.
I'm sorry,
تفضل دكتور رائد.
The voice, it's بس الصوت راح ما سمعتك. هل كان في سؤال أو لا؟
لا أبدا. دكتور عبد الله قال لك thank you very much. قال لك شكرا يعطيك العافية.
Thank you.
Thank you very much دكتور رائد يعطيك العافية. Our next presenter is Dr. Ahmed Rashid. Are you around, Dr. Ahmed? دكتور أحمد الراشد. أهلا وسهلا دكتور أحمد.
حياكم الله. هل في صوت؟
صوتك واضح بس في صدى شوي. تفضل حط سلايدات دكتور أحمد. أحسنت. Can you display it, دكتور؟
Yeah.
دكتور أحمد.
I can hear you.
Can you display the slides? سوي لهم عرض إذا ممكن.
I feel it, but I don't know.
ما تقدر تعرضهم من عندك؟ دكتور أحمد. سم.
ظهرت الآن؟
لا للأسف ما هي موجودة as a display، يعني كل الشرائح أمامنا.
للأسف ما أدري، أنا حاولت أكتب مرة.
دكتورة هند، دكتور عبد الله الثميري, is it okay if Dr. Ahmed present in this way? Can you see the slides?
Yeah, sure.
أنا ماني شايفة slides.
Okay, Dr. Ahmed, try to post the slides again.
أيوة، okay.
Go ahead, ممتاز، تفضل، بالتوفيق.
السلام عليكم. Thank you for giving me this opportunity to deliver this project under the name of severity of COVID-19 among Asian people in two Saudi public security hospital retrospective cohort study. So I have nothing to disclose, and COVID is a pandemic disease and infected more than 86 million and more than 1.8 died. And the severity of symptoms is varied from mild to critical. And there are a lot of risk factors that increase the severity of disease, like the age and obesity and comorbidities, and also some medications, for example, the ACE inhibitor. It has been hypothesized that exposing patients to ACE inhibitors increase the expression of ACE2 by negative feedback, and thus increase the binding of coronavirus to the epithelial cell. Subsequently, this will increase the development of severe disease. So with the current available data, there are lots of contradictory findings reported with the use of ACE inhibitor in different populations. So the use of ACE inhibitor use may suggest population difference and may play a role in disease severity. So the objective of the study is to investigate the influence of ACE inhibitor exposure and the severity of COVID during hospital admission in Saudi Arabia among overall population and high-risk populations. So it is retrospective study conducted in King Fahd Medical City and Prince Mohammed bin Abdulaziz Hospital. The IFP earned from both institutions. So the inclusion criteria: patient age more than 18 and period from April to June, and being treated with ACE inhibitor for 6 months before the admission. And also a patient diagnosed with COVID by PCR from the nasal swab, and also the exclusion criteria: pregnant women and incomplete medical record. So this is the definition of COVID severity: mild, moderate, severe, and critical, based on the WHO definition. So the study outcomes, we want to classify the COVID severity based on the WHO definition. Also need of ICU admission, also need of non-invasive ventilation, and need of mechanical ventilation and in-hospital death. So this is the study flow diagram. So we got 532 patients, and we exclude 178, and the remaining is 354 classified in ACE inhibitor users and non-ACE inhibitor users. With the multiple variable logistic regression we have done, and also we have done the propensity scoring, and we lost subjects in this situation, and we become with 60 to 63 and for non-ACE inhibitor users, and also 145 for ACE inhibitor users. So we use the R program for analyzing the data. And also continuous data were expressed with a mean and standard deviation and median with the interquartile range. And we have also checked the assumption by doing the QQ plot and also Shapiro test. We use a t-test for the normal distributed data, and also we have done the Mann-Whitney test for a non-normally or abnormally distributed data. Also, we have done the chi-square and also Fisher exact test. Also, we have done multiple logistic regression model adjusting for the confounder. So the confounder are age, also sex, BMI, diabetes, hypertension, renal failure, and number of comorbidities, and also the COVID treatment regimen. All statistical inference was drawn with 95% confidence interval with p-value less than 0.05. And also we have done the Perroni adjustment for the controlling or adjusting for the multiplicity or family-wise error to control the inflation of alpha-1 error. Also, we have done sensitivity analysis, both of them, the propensity score matching and also inverse propensity score weighing for a high alert, specifically for the high risk patient subject. I just want to show you, this is before doing the propensity score match. This is the data before adjustment, and after adjustment, see it's approximately, it's a kind of a mirror diagram. So this is before doing the propensity, and this is after adjusting for the confounder. So baseline characteristic. There at the beginning there is a difference in the age and also hypertension, diabetes, cardiovascular diseases, and also renal disease. But we have done adjustment for these confounders and covariates. So after doing adjustment for this confounder, we have seen the outcome. So the first one, we just want to see whether the patient who is receiving who is on ACE inhibitor compared to non-ACE inhibitor group. So we have seen that patients who's on ACE inhibitor have higher severe and critical case of COVID. The odds ratio approximately 8.25. So it's approximately 8-fold. P-value, we have done the p-value significantly, it is statistically significant. And also after adjustment for the confounder, we have done Perroni adjustment in order to control the family-wise error, and which was approximately, it is significant. Also, with the doing of propensity score matching, it was significant. And also we have done several adjustments in order to confirm that this was statistically different. So after propensity matching, score matching, and was significant, and also with the Perroni adjustment was significant. So the patient who on an ACE inhibitor was higher risk of developing severe and critical case. And also regarding to the ACE inhibitor, it was higher in patients on ACE inhibitor and also the mechanical non-invasive ventilation was higher, severe in ACE inhibitor users. On the other side, the mechanical ventilation and hospital death was not statistically different between the both groups. So there is no difference between ACE inhibitor and non-ACE inhibitor users. Let's go to the specific population, and we have done the inverse probability score weighing for the high-risk population, diabetes or hypertension or renal dialysis. The odds ratio one I've seen it is significant, and it was significant for the diabetes alone, and it was significant in hypertension alone. So the patients who have diabetes or hypertension or renal have and on ACE inhibitor develop higher risk of severe or critical case, except in patients who have diabetes plus hypertension. It was not significant. So the conclusion, the ACE inhibitor was used to find that it's associated with adverse effects on the severity of COVID disease in Saudi population, and high-risk population, diabetes, hypertension, renal, were higher risk of developing severe COVID disease as compared to non-ACE inhibitor group. Compared to non-ACE inhibitor user, the ACE inhibitor user have 8-fold, as we have seen in the table. It was 8-fold higher risk of developing critical or severe COVID, and approximately 7-fold higher risk of developing intensive care unit ICU admission, and also nearly 5-fold higher risk of requiring non-invasive ventilation. That means higher risk of oxygen demand. Also, patients with high diabetes and hypertension or renal have 5-fold higher risk of developing severe COVID disease. So this is the conclusion. If there is any question.
Yeah, شكرا دكتور يعطيك العافية. The idea is great. And it's very interesting, حقيقة, especially in the era of COVID. Can you hear me? Because I believe there's an echo.
Yes.
Okay. Yeah. But I do have a couple of questions. Not questions, some comments and a question. There's a 16 author in this publication. I understand it's a multi-center, but then do you actually need these 16 contributors? Maybe yes, but that's just my comment. My other questions would be, this is from my information. So what would be the difference, between using the PSM and IPSW, the inverse propensity score? And this is from my information. Do you know, or the biostatistician have done it? Why do we use actually propensity score? Instead of the usual regression. What's the difference? Does it make a difference? Is it related to your confounders, the number of them?
Yeah, so we have done the propensity score matching in order to avoid the bias selection, and also we have done adjustments for this in order to ensure that we have done it in an appropriate way. And then we have done also with the propensity score matching, we have done with the covariate age, comorbidities, and number of COVID regimen. So we have done it. The adjustment, I can show you the adjustment. I can take it here. We have done it with the R. Here. Yeah, see here, this is some log plot. It was done with propensity score matching model, and it is here. We have done it with adjustment caliber 0.1, and we have include the number of comorbidities, age, BMI, hypertension, and also that BMI, diabetes, and renal dialysis. So we include all these components in order to ensure that we have balanced the data sets and also to avoid the bias selection. You hear me?
Sorry, as I was unmuting myself, so I don't hear an echo. If you go back to the slide where your results for the, all of your confounders, and why did you put heart failure with CAD? Why didn't you use heart failure as a single independent category?
Yeah, we have fit several multiple models in order to ensure the appropriate model.
No, my question is, why didn't you include heart failure as separate? Why did you actually put it with CAD?
We just include all of the cardiovascular diseases and specific. We've done it in April, and there is no data about heart failure associated with the severity of COVID. So we just gather all the cardiovascular in one item.
Yeah, I know, but hypertension, you put it as a single entity. So why not heart failure? Mind you, majority of patients on ACE or ARB, definitely in a way or another, there will be heart failure. Majority, I'm not saying all, but that's true, because you want, if you see one of the slides where you find what the results, where you see it was actually statistically significant, patients with heart failure / CAD, but we really don't know, was it the CAD itself, or is it, no, it's not in the last slides. If you go to the slides, previous slides, please.
This one?
Here, I think, yes, the baseline. Oh, okay, so this is your, now go to the baseline characteristics, please. So if you see here, yes, so majority here of patients, I don't understand, is it heart failure or is it actually CAD? So you can't, probably should separate the two. CAD is not heart failure. Heart failure is a totally different entity, and that's just my recommendation for you. Although I think you probably publish it already, but it's a great job, a great piece of art. What would be your recommendation? My last comment, question. What would be your recommendation? Should we stop ACE?
In this situation, it is unclear picture until now, and we have 3 randomized control trials running in the world, one in US and one in Britain and one in Australia. So we are waiting these waiting the result of this randomized control trial in order to take this decision. But anyway, and this until now, sometimes of patients, for example, patient hemodynamic unstable in this situation, we can hold these antihypertensive medication and control it with other medications, and we can stop it only for example, 2 weeks or less. It's a kind of brilliant strategy in order to see further data available in there.
Okay, thank you, Doctor. Thank you so much.
سلام.
Appreciate it.
دكتور أحمد، معك دكتور عبد الله الثميري. سامعني دكتور أحمد؟
نعم.
ممتاز. So first of all, congratulations, another great project. صراحة, this project is really good, but maybe due to technical difficulty, it was challenging to deliver every aspect of it. However, I have a lot of comments, but I'm gonna deliver some of them here. Can you go to the inclusion and exclusion criteria, the diagram that you put, that you actually, this the process, yeah, in 7, yes. So here you have 73 people not completed because of incomplete file or missing file. Have you tried to actually go back and do like sensitivity analysis by doing like the certain statistical technique to address the missing files?
Yeah, this one.
No, yeah, number 7, slide 7. So you have the second part. You have you started with 532 patients, and due to incomplete file, you actually excluded 73. That's a lot. That's a big number.
Yeah.
So have you tried to do like statistical technique to address the missing values?
Yeah, there is an technique that can be used for controlling the missing data. For example, mice. In this situation, but we didn't use it, unfortunately. But I have in this project we didn't use, unfortunately. That's why we exclude these patients. But I think this is a good idea to use it in other projects. I've used this technique in other projects nowadays. So it's perfect. It comes with you can control these missing data. But unfortunately, I didn't do this in this project.
Yeah, I think there is always area of improvement, because this would be one of my biggest recommendations, is go back and address it, and probably it will improve your matching technique subsequently if you do that. Because if you're doing matching, you can do certain statistical techniques to help you out, improve your, So the second part, you mentioned in the method, when you started, that you did control for covariant and matching. But the results, when you presented two different models, one with covariant and regression, and the other one with the propensity score without controlling, right?
No, with controlling.
With controlling for the propensity score.
Yes.
And other covariant as well.
Sorry?
So you control for propensity score, and you also double control the covariance.
Adjustment.
Oh, double adjustment.
Yeah, it's kind of double adjustment.
Yeah, it's not recommended to do that, but I can get some of the researcher likes to do that in conservative. So, and the variables that you included in your propensity scores, what you can go back 2 slides, maybe the descriptive. Dr. Hind also asked you almost similar questions about that. Yeah, so no, slide number 10. Yeah, so you control for the one that came significant, or every, or clinical.
Also, we include also the clinical importance. For example, the obesity we included in the obesity matching score, but it wasn't significant.
And also gender, if I remember correctly, right? So one of the issues that you had here was the matching, that you dropped a lot of patients due to matching, and you did a conservative matching. So you adjusted your propensity score matching to 0.1, which is very, like, they have to match in 90%, okay? And you did double adjustment, and you control for non-significant. This very, very conservative approach. I would have, I would love to see it in a very, methodological approach, so we can see it in the original. Then you can do what you did as a sensitivity analysis, if you would like. Okay, so if you did the first methodology for what I'm suggesting, you would end up with more matched sample. It will give you a stronger evidence, and you can do what you did as a sensitivity analysis for adjustment. But thank you. It's a great project, great presentation. I enjoyed reading. Thank you.
Thank you, Dr. Ahmed. يعطيك العافية. Thank you, Doctor. Thank you, Dr. Hind. Thank you, Dr. Abdullah. We'll move now to the next keynote speaker, and then we'll proceed with the rest of the presentations.
We'll start the next session. We'll start the next session with our next distinguished speaker, Dr. Adel Al-Haraf. Dr. Adel served as the Vice President of the Drug Sector at the Saudi Food and Drug Authority. He received his PhD in Medicine and Therapeutics from the University of Glasgow. He joined the Saudi FDA in 2004 and was among the first team responsible for the establishment of most of the drug sector development and regulations, including pharmacovigilance, licensing, inspection, and drug evaluations. Dr. Al-Haraf is the Saudi FDA representative at the International Council for Harmonization, and he is the board member and head of Executive Committee of the Saudi Authority of Intellectual Properties. He is also a member of many steering committees, including the National Center for Complementary and Alternative Medicine and the National Diabetic Center. So let's welcome all Dr. Adel, and please start your presentation. Thank you so much.
بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. الصوت واضح؟
Yes, it's clear.
ممتاز. أول شيء حاب أعرف بنفسي. معكم أخوكم فيصل بن علي التغيم. أنا أحضر اليوم بالنيابة عن سعادة النائب دكتور عادل هرف لانشغاله يعني باجتماع طارئ. الصراحة هو حب ورغب جدا أنه يكون متواجد اليوم، ولكن للأسف اجتماع مفاجئ يعني منعه من ذلك، فأتمنى أن يكون كافي بالنيابة عنه. ما أدري ظهرت لكم السلايدات
لا.
أو
No,
طيب.
please, can you share again?
طيب كذا.
Yes.
طيب.
Thank you.
صراحة سعادة النائب بتشكر منكم على التنظيم الصراحة والتواصل الفعال من قبل المنسقين والمنظمين. الله يعطيكم العافية، وإن شاء الله اليوم أن يكون يعني كافي بالنيابة عن النائب الدكتور عادل والظرف اللي يعني والاجتماع المفاجئ اللي حصل. أول شيء يعني عشان تكون يعني عندكم صورة من المتحدث، أنا صيدلي أعمل في قسم التجارب السريرية كمقيم للدراسات السريرية. Okay. يلا بسم الله على بركة الله. This will be the outline of my presentation. Today we'll talk about a little bit about the history of clinical trials in Saudi Arabia, and then we'll talk about the clinical trial regulations. And since this is a pharmacy-oriented conference, we'll talk about the pharmacist's role in clinical trials. Then we'll talk about the status of clinical trials in Saudi Arabia and some statistics, and the Saudi FDA initiatives regarding this. In 2009, the clinical trial was a committee. The aim was to establish and to write guidelines to regulate clinical trials in Saudi Arabia. Then in 2010, the clinical trial department was established, and in April, it was mandatory to obtain FDA approval before conducting a clinical trial. We have an electronic system called the Saudi Clinical Trial Registry. It was launched in 2013. We also published our guideline, regulation, and requirements for conducting clinical trials on drug in 2015. And also regarding the GCP inspection, it was started in 2017. Before that, we did like a few number of GCP inspection visits, but officially it was in 2017. And we also have the Clinical Trial Scientific Consultation Committee, which started in 2017. This is some statistics about the committee. We have 39 physicians and 24 clinical pharmacists, 4 biostatisticians, and 4 epidemiologists. Okay, now we'll talk about the regulations in Saudi Arabia. All clinical trials involving drugs must be registered through the Saudi Clinical Trial Registry. And the applicant must adhere to the GCP, ICH GCP E6 guideline and the guideline of regulation and of research ethics code on living creature, and SFDA regulations and requirements for conducting clinical trials on drugs. Okay, regarding the phases of clinical trials, as you know, there is 4 phases. For the requirements for phase 4, it's mandatory to register the study in at the Saudi FDA, but the level of regulation is a notification only. No approval is needed, and the documents required from the applicant is the clinical trial protocol, the informed consent form, the IRB approval, and the statement of investigator. This statement of investigator is a form that the investigator fill, and he signs that he will follow the GCP and the Saudi FDA regulations and the guidelines. Regarding early phases clinical trials, it's mandatory to register at Saudi FDA, and it's mandatory to obtain SFDA approval before conducting the trial or initiating the trial. Regarding the reviewing timeline, for phase 2, it's 60 working days, and for phase 3, it's 30 working days. But we are working on shortening this time, and it will be official soon. It will be 40 working days for phase 2 and 20 working days for phase 3. This is a table for the requirements for early phases. You can find it at the SFDA website. I would like to mention it's the applicant can submit the documents as a soft copy. In the past, we required the hard copy, but now we ask for a soft copy, so the applicant can submit a CD or a flash drive. Regarding clinical trial amendments in early phases, as you know, some trials take 5 or 10 or even 20 years, so there will be a lot of amendments during this time. There is 2 types of amendments: a non-substantial amendment and substantial amendment. In case of non-substantial amendment, the SFDA should be notified through the annual progress report. In case of substantial amendment and addition of new site, the SFDA approval is mandatory. In case of urgent safety measures need to be taken to protect trial subjects from immediate hazards, the SFDA allows such actions to be taken without prior approval. But on the other hand, the SFDA must be notified as soon as possible. Okay, regarding adverse drug reaction, we have something called SUSAR, which is suspected unexpected serious adverse reactions. It's divided into 5 categories. Fatal and life-threatening should be reported to SFDA within 7 days. The hospitalization or significant disability or birth defect should be reported in 15 days in accordance with the ICH E2A pharmacovigilance guideline. We also have something called DSUR, which is Development Safety Update Report for investigational products. And the progress report can be submitted annually to the Clinical Trials Department. As you know, for investigational products, they are not marketed or available in Saudi Arabia. So in this case, mostly it will be imported from outside Saudi Arabia. So the applicant needs to fill the clearance form and submit it to SFDA in order to obtain the investigation product. In case of completion, termination, or suspension of the clinical trial, the applicant should inform SFDA within a maximum of 60 days with attached of a proof of IRB notification. And it's mandatory to submit the final report within 1 year in accordance with ICH E3 guideline. These are some forms we made to facilitate the applicant work, and the applicant can find it. It's electronic. The applicant can find it at the SFDA website, can fill it and sign it and submit it easily to SFDA. This is, as you can see, the importation request form, the statement of investigator form, the financial disclosure form, and the progress report form. We also have our guidelines in both languages, English and Arabic language, available at the Saudi FDA website. Okay, now we'll talk about the pharmacist role in clinical trials. I would like to mention that if I'm going to talk about the pharmacist role in clinical trials, I might take 1 or 2 hours. It's a huge part and a huge role. 2 or 3 slides won't explain a lot of the hard work pharmacists do, but I tried to summarize it for the sake of time as much as I can. The next information is like the most important. It's not all the information. So the pharmacist should review the clinical trial protocol before the start of the trial, and to make sure are all the contraindications listed in the inclusion criteria of the protocol, are all drug disease interactions listed in the exclusion criteria, have all expected side effects being included in protocol and informed consent form. And also the trial specific dispensing procedure. So in the clinical trial, the dispensing procedure may be different from the normal case. So the dispensing procedure should be checked and approved by the clinical trial pharmacist. Also, the staff training, the pharmacist should train his colleagues or the study or the research team regarding the investigational product and how to handle it. The pharmacist should be a part in initiation meeting with the trial sponsor or investigator to agree on the accountability log and other trial documents, to finalize arrangements for the investigational medicinal product delivery and receipt and storage, and discuss any special requirements for trial drug dispensing. The investigational product should be stored separately from the normal pharmacy stock in area with restricted or controlled access. It should not be stored on ward or clinic area unless it's used for emergency care. And it should has a label. The investigational product should be labeled and in certain criteria. And the most important thing to have a sentence for the clinical trial use only, and it should be separated from the other drugs in the normal pharmacy. The pharmacists also make sure to check regularly check the temperature monitor, and any deviation should be reported to the study monitor, and the medication should be quarantined. Also, the drug accountability, it's a task for the pharmacist to make sure of the accountability of the drug, how many, in case of pills or tablets, how many prescribed or dispensed, how many returned or left over, how many expired, and at the end of the study, is the remaining or excess drugs will be destroyed or sent back to the sponsor. Also, the pharmacist should keep records and update the pharmacy file. Okay, now we'll talk about some statistics regarding clinical trials in Saudi Arabia. These are the numbers of clinical trials from 2009 until 2020. And the blue color is the early phases, and the orange is phase 4. As you can see, in 2020, the number of clinical trials increased because of COVID-19. A lot of researchers and sponsors would like to conduct clinical trials on COVID-19. Also, this figure is for clinical trials and their distribution by phases. So we have 42% phase 3 and 42% phase 4 clinical trials, 10% as a phase 2, and 4% as bioequivalence studies, and 2% as a phase 1. Regarding early phases, you can see 50% of the early phases in Saudi Arabia are ongoing. 14% are completed. We have also 7% as rejected. For the type of submission, if it's a commercial or non-commercial, sponsored or investigator initiated, we have commercial 300 commercial clinical trials and 127 non-commercial clinical trials. Okay, now we'll talk about some statistics in 2020 only. The number of clinical trials evaluated and reviewed by our department is 61 clinical trial. and number of COVID-19 clinical trials is 17, and the number of GCP inspections is 8, but as you can see, it's a low number. We had this GCP inspection plan for 2020, but COVID-19 have interrupted our plans. That's why the number is very low. I would like to mention here, in addition to these 8, we did conducted a GCP inspection in China and in Russia for the vaccines. Also, we have received 2,689 queries or consultations via emails and phone calls. This is the published list for clinical trials in Saudi Arabia. You can find it at FDA website. You can find different information about the clinical trials, the title, the trial phase, the protocol number, the drug, and the sponsor. Also, you can find in which sites are conducted in Saudi Arabia. Okay, now we'll talk about Saudi FDA initiatives. We have published well-established regulations and guidelines to make it easier for the applicants to apply for clinical trials. We have reduced our reviewing timelines. In the past, it was to 120 working days for phase 2 and 90 working days for phase 3. Now it's 60 working days and 30 working days. And in the future, inshallah, it will be 40 working days for phase 2 and 20 working days for phase 3. We have allowed parallel submissions, which means the applicant can submit to the IRB and SFDA at the same time to prevent any delay, because some of IRB committees or ethics committee take a long time. We have mandatory GCP training for investigators to increase the quality of research. We have also GCP inspection to make sure that clinical trials are conducted in accordance with the GCP. We have also made workshops with our partners at the National Committee of Bioethics and SAGIA, which is the Ministry of Investments now. And we also have worked on the CRO project. The CRO is the Contract Research Organization. We have local CROs in Saudi Arabia. Any sponsor or researcher can outsource for CROs. We have worked at the project to make the CRO environment much better and to facilitate their work. And we'd like to talk about the phase 1 clinical trial guideline. In the past, phase 1 clinical trials, it was not allowed to be conducted in Saudi Arabia. Now I'm happy to say we published the phase 1 guideline, I think in November. But I want to make it clear, there is 2 parts of the clinical trial, of the phase 1, the clinical trial and the unit itself. So we published the clinical trial phase 1 guideline, and we are working on the phase 1 unit guideline for licensing and accreditation. And now, as far as I know, there is only one unit, phase 1 unit, allowed to be conducted in, which is at KMARC, and it will be dealt with the case by case until we publish the second guideline. We also did an awareness campaign in the past, which took place in 6 different cities in Saudi Arabia: Riyadh, Jeddah, Mecca, Al Madinah, Dammam, and Abha. We also did a video for the public, which was published in social media and in Snapchat and Twitter, and to increase awareness in the public. Thank you so much. This is the end of my presentation.
Thank you, Dr. Faisal, for this excellent presentation. I have one question from Dr. Khairi. He said, do you think that the outcome of the study in terms of severity and intensity of the side effect will be similar to other GCC countries or other European countries, or they will be other outcome?
This question is for me?
Yes.
Sorry, can you repeat the question, please?
Right. Do you think that the outcome of a study in terms of severity and intensity of the side effects will be similar to the other GCC countries or other Europeans?
Actually, I don't know what happens in the GCC countries. I cannot compare us to them. I'm not familiar with their regulations.
Sure. I have a question, if you don't mind. In
My
the
pleasure.
first talk, our other distinguished speaker, Dr. Majid Al-Jaisi, he said something about there is an increment in research in the region about like 34%, which is an excellent result. From the Saudi FDA perspective, what are the challenges that the researcher faces to do a research?
Okay, from my experience, unfortunately, some of the researchers in Saudi Arabia, I want to be clear, we are talking now about investigator-initiated trials, not sponsored clinical trials. The investigators, unfortunately, he will be very well known in his field, and it will be like the best cardiologist or best neurologist in the Middle East. But unfortunately, in terms of clinical research, he will have less skills, which will make a lot of difficulties for him to conduct clinical trials. Also, another challenge is the IRB reviewing timelines. Some IRB committees take very long time to give their opinion or to give their approval or rejection. Some IRBs take 6, 7, 8 months. Now in COVID-19, the case is different because COVID-19 clinical trials has a priority. So I'm talking in the normal situation, some IRBs take 6, 7, 8 months. Then the applicant or the sponsor just will terminate the study and decide not to conduct the study.
Okay, it's clear. May I ask another question, please?
Yes, please.
I think one of the slides, it mentioned that in case of ADR leading to a mortality, the submission should be within 7 days. Am I right?
Yes, this one, yes.
So let's say that the submission happened after 7 days. Will you accept, I mean, as an authority, will you accept the ADR?
Yes, of course, but we will ask the applicant why the delay, because this happens sometimes. We get the reports after the timeline, but the applicant says, for example, I will give you one case. The patient is hospitalized and he did not inform the study team. He is hospitalized in a different hospital. And after 20 days or 30 days, the study team finds out that this patient was hospitalized. The study team informs this day at the same day. So the study team says, we informed you at the same day. We just known this participant was hospitalized. It happens sometimes, but we just ask why is the delay, so to make sure there is no deviations or doing wrong from the study team part. That's it.
We are very thankful, Dr. We are very thankful for Dr. Adel Al-Harf, and we are very thankful for you, Dr. Faisal, for joining us. And يعطيك ألف عافية ما قصرت.
الله يعافيك، هذا واجبنا. والدكتور عادل صراحة رغب جدا إنه يكون موجود، لكن للأسف ما الله كتب، وإن شاء الله يعني يكون كفينا وأدينا الواجب يعني.
ما قصرت. Thank you so much. Now we'll move to the sessions. Our judges for the sessions they are the same judges. Dr. Aus Shamsan, he's a professor and the current dean of the College of Pharmacy at King Saud University and the chair of the Future and Health Task Force in the S20 Engagement Group, and he is the editor-in-chief of the Saudi Pharmaceutical Journal. The next judge is Dr. Thamer Shamri. Dr. Thamer is an associate professor of health outcomes at the College of Medical Applied Science and the College of Pharmacy at Riyadh Ilm University. He is a pharmacoepidemiology and pharmacovigilance consultant and serves as the director of the National Pharmacovigilance and Drug Safety Center at the Saudi FDA. The third judge is also another figure, Dr. Abdullah Thmeri. He's an assistant professor at the Department of Clinical Pharmacy, College of Pharmacy at Prince Sultan bin Abdulaziz University. He has a PhD in pharmacoeconomics and outcome of research, and he's an active member in the International Society of Pharmacoeconomics and Outcomes Research since 2011. And the last judge, our colleague, one of my mentors, Dr. Hind Al-Mudemir. She's a clinical pharmacy consultant in cardiac science at King Abdulaziz Medical City in Riyadh, and she is a program director of the Specialized Cardiology Residency Program and an associate professor of pharmacy practice at the College of Pharmacy at King Saud bin Abdulaziz University for Health Science. So, thank you so much, judges. We'll start with the first speaker, Dr. Khazra Sultana. She's going to present a topic entitled A New Model to Predict Bleeding Score in Non-Valvular Atrial Fibrillation Patient Using Novel Oral Anticoagulant in the Arab Population. Go ahead, Dr. Sultana.
Assalamu alaikum. Can you hear me?
Yes.
And yes. Can you see my slides?
Now we do.
Okay, just one second. Okay, is that clear?
Yes, thank you.
Okay. Assalamu alaikum everyone. I'm Ms. Khazra Sultana, and I thank the Pharmacy Research Committee for giving me this opportunity to present my study, a new model to predict bleeding risk score in non-valvular atrial fibrillation patients using novel oral anticoagulants in the Arab population. So the outline for my presentation would be, first we will have a little background about the existing models, the aim of our study, the methods used, the results, and finally the conclusion from our study. So non-valvular atrial fibrillation is considered the most common supraventricular arrhythmia. AF patients are at a high risk of developing ischemic stroke by 5-folds and systemic thromboembolic events compared to non-AF patients. Thus, they require anticoagulation therapy to prevent such risks, but prescribing anticoagulants must balance both the embolic as well as the bleeding risk. To decide which patients with non-valvular AF are likely to benefit from anticoagulation, a validated bleeding risk score needs to be calculated. Widely used scores known to calculate the stroke and bleeding risk are the CHAD-VAS score and the Hasbled score. Since the CHAD-VAS score is used to provide the stroke risk and the Hasbled score provides the bleeding risk. The variables that are considered in Hasbled model to calculate the score are usually the hypertension, abnormal liver and renal function, stroke, previous bleeding, labile INRs, elderly age, drugs, and alcohol. Now, there are other models in the literature like the Kuger, Caron, Schreman, Atria, Hemorrhages, Reti, Orbit, and Garfield, which have evaluated the risk of bleeding with the anticoagulant warfarin. Based on the available evidence, there are lack of studies for predicting the bleeding risk score for the direct oral anticoagulants like dabigatran, rivaroxaban, apixaban, and edoxaban. This cohort study aimed to develop a new validated bleeding risk prediction score with the use of direct oral anticoagulants in patients with non-valvular AF in the Arab population. So this is a retrospective cohort study. We had a total of 1,722 patients, out of which 829 were collected from King Abdulaziz Medical City, 115 were from King Fahd Cardiac Center, 778 belong to the Prince Sultan Cardiac Center, which are all tertiary care hospitals. The inclusion criteria for the patients were those who were greater than or equal to 18 years, belonging to both genders, prescribed direct oral anticoagulants for a minimum of 1 year, and those patients diagnosed with non-valvular AF. These patients also had one or more of the following comorbid conditions like cardiovascular disease, diastolic or systolic blood pressure, coronary artery disease, diabetes mellitus, malignancy, specifically gastrointestinal, stroke or transient ischemic attack, thyroid disorder, liver disease. So the outcome for our study was the prediction of a bleeding risk in non-valvular AF patients who are likely to benefit from prescribing direct oral anticoagulants at the onset of anticoagulation therapy. Bleeding was defined as a history of blood transfusion within 3 months from starting the direct oral anticoagulant, or a history of drop in hemoglobin more than 2 grams within 3 months from starting the direct oral anticoagulant. So these were the following variables that are considered as the predictors of bleeding: gender, age, concomitant drugs like aspirin and clopidogrel, uncontrolled blood pressure, especially 150 above 150 systolic, liver failure related with high AST or ALT enzymes, which is greater than 2 times or 3 times the upper standard limit, renal failure divided into 5 stages, and dialysis according to the KDIGO guidelines. The liver diseases like hepatitis, cirrhosis, hepatic cell carcinoma, and other types of liver diseases. Central nervous system diseases like stroke, TIA, transient ischemic attack, and other CNS diseases, and of course, atrial fibrillation. Now, the analysis that we used for developing the model was first, the analysis used a resampling approach for variable selection. During the analysis, 75% of the data is chosen randomly for model building and 25% for validation. The backward elimination logistic regression method was used to build the model. The process was repeated for 1,000 times with different selection and validation samples. The variables that were selected at least 100 times were included in the final model. So after the model, finalizing the variables to be included, we used a 5-fold cross-validation to assess the model fit and misclassification probabilities for the variables. The logistic regression using a penalized maximum likelihood estimation method, also called as FERT penalty, was used to fit the model for cross-validation. The discriminative ability of the model was measured by the concordance statistic or area under the curve and receiver operator characteristic curves for assessing the classification capability of the validated model for the validation model. The value of the C statistic, if it is 0.5, it means no discrimination, and if it is 1, it means perfect discrimination. So if it is between 5 to less than 7, poor discrimination, between 0.7 to 0.8, acceptable, and so on. For the risk prediction tool, we used the penalized likelihood parameters for its development, and the accuracy of the prediction tool is reported using sensitivity, specificity, and Breyer score. So these are the results. So out of our total sample size, we had 192 bleeding events, out of which 42 were blood transfusions and 94 had a hemoglobin drop of more than 2 grams. We had 10 patients with missing data where it was not clear that what type of bleeding event happened in them. So these are the parameter estimates, standard error, and corresponding p-values of the final model. So gender, age, the last direct oral anticoagulant that was used, and other comorbidities, and so on. So this is the ROC curve for the 5-fold cross-validation, and the ROC curves will give insight to the model discrimination performance. This allows for determining the optimal sensitivity and specificity pair of the model. So this is the area under the curve estimate for the 5-fold cross-validation, and the results indicate that the model is very stable, and the average AUC or the C statistic of the validation is 0.749 with a p-value less than 0.05. The Breyer score estimate for the predictive accuracy of the model is 0.095. We have fixed a cut probability or value of 0.12 for the logistic regression equation with 72.7% sensitivity and 66.3% specificity for the risk prediction tool. So this is a screenshot of the Excel, because the logistic regression equation was converted into an Excel score calculator, and this uses a simple interface to enter the patient data. So the patient data can be entered here and produces an instant risk score with a green color code meaning safe, yellow moderate or borderline risk, and red to be highly risky. So these 15 variables were considered and scored according to the binary scale to predict bleeding risk. Later at the end of the presentation, I will show you the Excel score calculator. So it does not require genetic variables and laboratory data. This model does not include the liable INR profiles of the patients. There were certain limitations to our study. This model cannot predict clinically relevant or non-major bleeding and non-major bleeding events. This score applies to people to those people only who do not consume alcohol. We were not able to do the follow-up of our patients. So further research is required to compare our model with existing models proposed for bleeding risk prediction with direct oral anticoagulants to evaluate its performance. So in conclusion, in this study, in a cohort of Saudi patients with non-valvular AF who are prescribed direct oral anticoagulants, we have identified 15 predictors of bleeding. This model provides the basis for use all over KSA and in the Arab world. This easy to use bleeding risk score will allow the clinician to quickly classify patients according to their risk category and pay closer attention and follow up for high-risk patients without laboratory and radiological monitoring. So I would like to thank the PI of the study, Dr. Mahal Ammari, and all other team members, and a special thank to our statistical expert, Dr. Abin Thomas. Thank you very much. Hello?
Yes, we hear you, Sultana. Thank you so much. It's a very interesting tool, especially these with the DUACs, and this is actually something new. I have very quick questions. First of all, so why did you go with a new innovative idea instead of, using the common ones, the common, bleeding risk predictors that I commonly use with the VKAs, and just use it with the DUACs?
So the thing is that normally we use the Hasbled score, correct? Which is for the warfarin. And there have been studies which have tried to use the Hasbled score with direct oral anticoagulants. But the problem is that these studies, if we use this score, they are not able to predict the discriminative ability of the model. Like for one of the studies, it only had the discriminative ability, that is the C statistic of only 0.68, which shows very poor prediction ability. And in many other studies, they were not even able to report this score. So that's one thing. And the other thing, as you know, that in the Hasbled score, it is also applied. There is a score that is given for people who consume alcohol. And most of the people in the Arab world don't consume alcohol. So that kind of does not give us perfect information for calculating the risk.
Okay, my other question is, did you try to compare your results to Hasbled score, for example?
Yes, that's a good question. We did not have the present enough information to compare our results with the Hasbled score. So this is the next stage of our study. We have just completed this, and we will try to compare our results and inshallah publish it.
Great, thank you. Go ahead and publish it, definitely. Probably we can pilot test that, even here and see.
Sure.
Thank you.
It's already accepted for publication in +1. We are just doing the revisions and then we'll submit it, inshallah, very soon.
All right, thank you. Thanks.
Thank you.
Thank you, Khadra, mashallah. Great study with great sample size, mashallah. But I just want to ask a quick question. Have you did a sensitivity analysis in the different sites that you applied, analyzing different sites?
Doctor, we used patients from different hospitals, correct? We used that as a combined sample. We did not separate the sample into different sites. So
no, I mean internally. I get the purpose of the study is just internally to see if the.
No, doctor, we didn't. we didn't.
Okay. Thank you. Thank you.
Thank you. So can I just show how this tool works?
Sorry, Dr. Sultan, we don't have much time, so we
Time?
have
Okay,
to move to
fine.
the next
No
debate.
issue.
Thank you so much.
Okay. Okay, great.
So the next speaker is Dr. Mohammed Zahani. He's going to talk about the evaluation of antimicrobial susceptibility of uropathogen isolated from pediatric patient in tertiary hospital at Al-Baha region of Saudi Arabia. So Mohammed, please share your slide and go ahead.
دقيقة بالله. واضح يا دكتور؟
اي تفضل.
بسم الله الرحمن الرحيم. Evaluation of antimicrobial susceptibility of uropathogens isolated from pediatric patients in tertiary hospital at Al-Baha region. Urinary tract infections is a bacterial infectious process affecting any part of the urinary tract in the bladder, urethra, or kidney. UTI is common in children and is one of the leading cause of hospital admission in pediatric population. So symptoms usually from the urinary urgency and frequency, as well as burning sensation during urination or lower abdominal discomfort and turbid urine. Most common the uropathogens is E. coli, Proteus, Staphylococcus saprophyticus, Klebsiella, and other enterobacteriaceae. The most common prescribed antibiotic are cotrimoxazole, nitrofurantoin, ciprofloxacin, and ampicillin. Significant relationship between the extensive antimicrobial use and its resistance and limited treatment options. The aim of this study is describe the antimicrobial resistance pattern of urinary pathogen isolated from pediatric patients. This study is a retrospective study. Was performed for patients who were admitted to pediatric department with the diagnosis of UTI and the large tertiary hospital, King Fahd Hospital at Al-Baha, from May 2017 to April 2018. The sample size, 118 patients, included all the patients between 0 to 14 years, excluded the patient from above 14 years old. The statistic analysis data were analyzed using SPSS software version 16, as well as data were entered into the database designed using the Excel spreadsheet and analyzed using the statistic this SPSS. The result from the 118 urinary bacterial samples isolated and identified, E. coli was the main etiologic agent. This in community hospital and in the community and the hospital-acquired infection. In the paragraph number 1, we found the E. coli, the 44%, followed by extended spectrum beta-hactamase E. coli, 11%, then Klebsiella pneumonia, 9%. The majority of the bacteria were isolated from females, 68.64%, while the remaining 31.36% were from the male. We found in the summer was at higher than in the winter, 58% in the summer, comparing with the winter 41%. Usually, in the summer, the methicillin sensitive resistant Staphylococcus aureus, MRSA, the most common, 100% in the summer, followed by E. coli, 65.38%. When you check in the winter, also the MRSA, the higher than other bacteria in the winter, 33, 33.33, fluid by coagulase negative by 12%. The antibiotic prescribed for UTI in this study, we found the nitrofurantoin 19%, trimethoprim sulfamethoxazole 16%, then augmentin is most common used. There is some medication less common used, for example, imipenem and amikacin. Imipenem as the restricted medication and amikacin for the highly nephrotoxicity. Amoxicillin, ampicillin, cefepime, this highly resistant. Antibacterial susceptibility pattern, the E. coli sensitivity for the last line indication is usually colistin, tigacycline, but tigacycline not used in the clinical because it's distributed into the tissue. Third generation cephalosporin 100%, then nitrofurantoin 95%. The resistance medication, cephalothene, this is represented in the lab for first generation cephalosporin. Ampicillin also 94% as highly resistant. Augmentin and cotrimoxazole have less sensitivity pattern against E. coli. Extended spectrum beta-lactamase positive E. coli, sensitivity for carbapenem, nitrofurantoin, norfloxacin 100%, as well as the augmentin and cotrimoxazole. This is usually used as empirical medication. Resistance for most of the cephalosporin, first generation, second generation, third generation, as well as fourth generation cefepime, levofloxacin. This highly resistant. Ciprofloxacin, gentamicin, except the lower susceptibility pattern to extended spectrum positive for E. coli. Klebsiella pneumonia. This is sensitive for amikacin. All the aminoglycoside we can use alone for UTI, comparing with other cases, should be given as combination. All the cephalosporin, ceftriaxone, cefepime, we can this has good sensitivity. If the medication less than 60%, we don't recommend to use. Augmentin is 50%. Well, we don't recommend to use as sensitive as the intermediate. For resistance, ampicillin, ciprofloxacin, cefuroxime, all this 100%, as well as tobramycin. At the same time, cotrimoxazole showed laser sensitivity pattern of 63%. For Enterococcus faecalis as gram-positive, the sensitivity for first generation cephalosporin, ciprofloxacin, linezolid, moxifloxacin, as well as vancomycin, but this is restricted for MRCA. Broad spectrum imipenem, this 66%. We don't recommend to more use for this because it maybe lead to increase the resistance in the future. Resistance usually cefepime for this fourth generation and tetracycline 100%, as well as gentamicin. This cholesterol heat map usually red color, this highly or maximum sensitive. For example, amikacin red color, we can cover most common of the antibiotic. The gray color is we don't have data. When you check for the blue color, this is lowest sensitivity. For example, cephalothene or a first generation cephalosporin with most common of the bacteria, except with Enterococcus faecalis. All this heat map will link between the type of the bacterial and the antibiotic. The yellow color is intermediate medication. For discussion, European Associated Urology Guidelines on the pediatric urology, UTI represent the most common bacterial infection in the children. E. coli 75%, then Klebsiella pneumonia, followed by Enterobacter. For the therapy for UTI in infant and children, for this guideline, usually recommended to use cephalosporin general, trimethoprim, sulfamethoxazole, ampicillin, amoxicillin, or augmentin. In our study, high prevalence of UTI in female 68%, then in the male 31.36%. Were observed, which correlated with other author in Saudi Arabia. The predominant isolated identified in our study, the E. coli 44%, followed by the extended spectrum beta-lactamase 26.92%, followed by Klebsiella pneumonia 9.32%. We found that E. coli and Klebsiella were resistant to cotrimoxazole, ampicillin, augmentin, is matching with other studies in Saudi Arabia. In this study, also the antibiotic susceptibility test of the extended spectrum beta-lactamase positive E. coli showed 100% resistance to most common of the cephalosporin. And the previous study conducted in other place outside of Saudi Arabia, this observed similar pattern of resistance for most common similar medication. Carbamidin, this broad spectrum restricted medication with us, highly sensitive for extended spectrum beta-lactamase, as well as amikacin 92.31%. This usually used for UTI as alone. We can use alone. In this present study, Enterococcus faecalis was found to be highly sensitive to linezolid, nitroglycerin, and vancomycin, but usually this is reserved for MRSA. The first choice or empirical for UTI are this conclusion. The first choice empirical antibacterial for UTI are nitrofurantoin, cotrimoxazole. In exception cases, oral cephalosporin can be used. We recommend use for bilateral or cephalosporin for bilateral, oral cephalosporin, nitrofurantoin, cotrimoxazole, augmentin, ciprofloxacin, gentamicin, and tobramycin. Current study showed us that nitrofurantoin 90% is most prescribed medication in inpatient and outpatient, followed by cotrimoxazole, then augmentin, cefuroxime 10%. Sometimes there is wrong in the practice for the azithromycin. We found the azithromycin also prescribed, while amikacin, amoxicillin, cefiblium, ampicillin, all this less common used because high rate of resistance or restricted medication. Recommendation: instead of following universal recommendation, current awareness of the antibiotic resistance trend of the uropathogens in the particular geographic location is significant consideration for selecting effective antimicrobial care. This reference. Thank you for allowing to share with you in this
يعطيك العافية.
conference. Thank you.
شكرا جزيلا. So in a nutshell, بس بختصر for the time sake, please. So now, what do you mean by not using the universal? Do you mean to have, like, each institution should have their own, antibiogram and then antibiotic recommendations for UTI in children?
We recommend to use according to the antibiogram. Usually in this hospital, we depend on the antibiogram for every 3 months or every 6 months sometimes.
Okay, and it seems to be effective?
Yes.
Okay, thank you.
Welcome.
Doctor, have you tried to conduct similar study in a different site, maybe?
Again?
Similar prospective study in different sites, just to.
For Nepal, this most common is switch with us in the result. We found this. Also in the King Salman armed force, also we found this is most common of the result for our study is similar. Okay.
Okay, thank you.
In our location, many studies also.
Okay, if there are no more questions, we'll move to the next speaker. Thank you, Dr. Muhammad.
Welcome.
Next speaker is Dr. Mahal Mleiki. He's going to present prescribing errors in virtually operated ambulatory clinic during the pandemic of coronavirus disease. تفضل دكتور Mahal.
Thank you, Dr. Khalifa. Is it clear, my presentation?
أيوة تفضل دكتورة. نعم واضحة.
So I'm going to present prescribing errors in virtually operated ambulatory care clinics during the pandemic of coronavirus. My outlines are going to be background, methods, results, conclusion. Prescribing errors are prevalent in healthcare setting and pose also a threat for ambulatory care patients. There is also an aspect of prescribing errors were explored during virtual consultation at tertiary care academic center, serving a diverse population. Our aim is to determine the occurrence and nature of prescribing errors for the ambulatory population who had scheduled virtual visits. So it's a retrospective study in a tertiary care institution in Saudi Arabia. We retrieved all electronic prescriptions from all the virtual clinics from March 22nd last year to April 30th last year. We included all the prescriptions for adult and pediatrics patients. We did a mean standard deviation for the descriptive variables, and the chi-square test was applied to determine the association of prescribing errors with prescriber age groups, and also association between types of prescribing errors and study variables. We excluded onco clinics, hemodialysis units, organ stem cell transplant clinics, and the obstetric as well, because they've been excluded from virtual clinics. A total of 310 prescriptions were included from the electronic database of virtual outpatient clinics. The majority of 217 patients were at the age group of more than 18-year-old, and the remaining 93 were patients under the age of 18-year-old. It also shows you the prevalence of comorbidities of the patients that varied from 1.1% to 58.5%. Diabetes has the highest prevalence with 70.3%, followed by hypertension, 52.7%, and pulmonary disease, 41.8%. As a total of 310 prescriptions, 232 without any prescribing errors, and the remaining 78 with 25% were there were errors. So the most common type of prescribing errors goes to the wrong drug with 52.5%, followed by wrong dose with 16.6%, and then wrong duration with 12.8%. In terms of medication categories, antacids and antiplatelet and antidiabetics as well took the most common medication categories with 46.2% antacids and 12.8% antiplatelet and 10.2% for antidiabetics. This chart can illustrate for you the number of items per prescription. The most items numbers were 2 to 4 medications per prescriptions, goes with 33%, followed by single medication 28.7%, more than 7 medications 22.3%, and then 5 to 7 medications with 15.8%. I have to mention that the consultants made more prescribing errors to the for the prescriptions with 28.5%. We did another subset category regarding the association between types of prescribing errors and each variable. Starting with prescriber type, as I said, consultant had the highest percentage, and then we subset each error according if it's a wrong dose or a wrong drug or wrong duration, and it came up. We found that 70%, the highest percentage, goes with wrong duration. In the other arm, for the medication categories, also antacids had the highest percentage with 80.5%. And then after that, antidiabetics with 46.2%, and then antiplatelet with 40%. As a conclusion, there was a 25% prescribing errors occurred during virtual ambulatory care clinics. Wrong drug, wrong dose were the two most common types of errors encounters. There is a need to develop strategies for reducing the occurrence of prescribing errors and improve ambulatory care patient safety. Thank you.
Thank you so much. That's actually interesting findings. Not interesting findings because we know this, but it is actually interesting study. My question is, when you collected the data, did you only use, the SRS, or you've used the TID?
We did review the TID for the clinical pharmacists, and as well for the pharmacists that did any changes for any prescription have been reviewed.
You see, my problem with relying only on the TID is that the TID, it has to be evaluated, because sometimes, because all of these errors, you have to go and see exactly what, because not every TID is an error. Because sometimes you will see the TID if they change the rate, for example, or the drug is not available, it will be actually written as a TID. So did you look this into consideration before you submit it as an error? Like, did you see the actual documentation for that?
Yes, absolutely. Before collecting it, where we know what is specifically the TID written for, either according to the types of error, either wrong drug, wrong duration, or wrong dose. So any other than that, like instruction from translating English to Arabic, were excluded also as well.
Okay, thank you.
Sure.
Dr. Amaha, congratulations for the project. It's very interesting, as I had mentioned before. But I have a comment. I wish you should explicitly explain how you define each error. We know that the title of the error, but we can do it in different ways. Like prescription error could be various of methods. There are a lot of definition when it comes to drug safety. So I wish you shared with us how you define each one of them. The other thing, the sample size was small because of the short time, and I understand that perspective. But if I had similar issue, I would probably reclassify some, improve the groups comparison. So the statistical in some of the groups.
Sorry,
Okay, sorry. So the reclassify some of the groups.
Now we cannot hear you.
Yeah, the sample size is small, so consider reclassifying some of the groups.
In terms of what do you mean, doctor? In another perspective, you mean? Or another?
No. If you go back to slide 9, for example.
That one, is it here?
Or 8, maybe is easier to explain. 8, slide 8.
Sure. Yeah, that one. Items.
Yeah, with you compare the consultant, the specialty, you could use different classification. For example, consultant, non-consultant, rather than having a group with 0%.
Yeah, we've
The
did that.
value means nothing.
We did that.
Yeah, that's that.
Yeah, we've did that. And
Yeah.
but
So
what I can't hear you. But if I understood your question, doctor, we've did that. We classified the prescriber type. Hello?
Yes.
نسمعك
Yeah.
تفضلي.
If it's consultant, fellowship, residence, associate consultant, assistant consultant, all of these were classified in our study as well. If I understood your question.
دكتورة مها أعتذر منك. I have to stop now. شكرا دكتورة هند، شكرا دكتور عبد الله، شكرا لحضوركم معنا، شكرا دكتورة مها.
يعطيكم العافية. شكرا.
الله يعافيك. دكتور ثامر، دكتور أوس موجودين معنا؟
موجودين.
أنا موجود. حياك الله.
حياكم الله جميعا. طيب we'll go to the next speaker, دكتورة سجل مزروع. She's going to talk about barriers and facilitators to conduct economic evaluation studies of the GCC countries, a survey of researchers. تفضلي دكتورة سجل.
السلام عليكم جميعا.
عليكم السلام.
اسف ماني قادر اسوي شيرنج للسلايدز.
You cannot share the slides?
لا. ما ادري هي فاتحة عندكم الان ولا لا.
No, we don't see the slides. She cannot share.
لحظة. السلايدز ما تفتح يعني ما تظهر لي.
Hold on, Doctor. I'll try to from our side. دكتورة؟
نعم.
Can you see it? We are sharing your slides.
بصراحة لا.
You cannot see it from your side?
لا.
Hold on.
الحين طلعت.
Okay, go ahead please عشان التايم. تفضلي.
طيب أوكي. السلام عليكم جميعا.
السلام.
Today I'll be presenting barriers and facilitators for conducting economic evaluation in the GCC countries, and this study was done from the researcher perspective. Next, please. Can I move the slides, or you will be moving the slides? Okay. So before going into studying or assessing barriers and facilitators, I want to explain the concept of economic evaluation and why it is important. So economic evaluation is one of the methodologies that's used to compare between two different interventions, healthcare interventions. And what do we mean by healthcare interventions? It can be either drugs, diagnostics, clinics, any healthcare intervention. So we compare these two interventions in terms of costs and in terms of outcomes. And also costs and outcomes are also diverse. So cost can be either direct cost, direct medical cost, which is medications, the cost of medication, the cost of hospitalization. And it can be also, it can goes beyond direct costs. It can include indirect costs, such as premature death and early retirement and any other associated costs that can affect the whole society. So when it comes to why there's an interest in economic evaluation, because it can improve the efficiency by optimizing resources allocation. Can you go back to the previous slide, please? The second thing is it can reduce the variation in health services to ensure equity. So people should receive the same amount of care, whether they are in cities or in rural villages. It can be also used in formulary decisions and price negotiation. Next, please. So economic evaluation is used widely in the Western countries such as the United States and the United Kingdom. However, in the GCC, the situation is quite different. So according to one of the recently published systematic reviews in 2018, they found that the total number of published economic evaluation in the GCC was around 49 studies only. And 40 studies, which is 80% of them, were partial economic evaluation. And what do we mean by partial economic evaluation? It is a very simple costing studies. We also called it drug utilization studies. Basically, you collect the amount of medication consumed by the patient and you multiply it by the unit cost, and that's it. So from this systematic review and other systematic reviews, there is a clear need. There is a clear need to enhance using or publishing economic evaluation. So we have a limitation in terms of number, and we have a limitation in terms of quality. In GCC countries, in Saudi Arabia and in other GCC countries, all aim to reach the financial sustainability in our healthcare sector. So one of the Saudi national transformational vision is to secure financial sustainability, and this can be achieved by multiple techniques. One of them is economic evaluation. So therefore, our aim next. Our aim is to assess the researcher barriers and facilitators, because researchers are the main drivers for publishing and conducting economic evaluation, and we thought that our assessing their barrier facilitators from their perspective is warranted. Next. So this is a cross-sectional study of health economic experts working in the GCC. This was conducted last year. So we used the electronic survey in order to reach all of our participants. The study was approved by the King Saud Medical City IRBFX. Next. So the most challenging part in this study is to identify experts. First of all, we used a couple of systematic reviews, the one I just mentioned and another one, and we looked at the reference list in these systematic reviews. And then we identify all authors and trying to get their emails as well. And because we believe that there are many health economists who are qualified, but they were not able to publish an economic evaluation, we also looked at the local health economic conferences speakers. So we identify a few conferences and we identify some speakers. And then finally, we asked the experts from the two previous stages to nominate or also to suggest their colleagues or anyone they been work with to suggest their names so that we can also include them in our sample. So the total was 143 eligible participants. Next. So in order to develop our tool, we couldn't find a tool that addresses all of our objectives. So we start looking at similar studies to conduct economic evaluation in decision-making process. We also looked at barriers and facilitators for conducting RCTs and research in general, because economic evaluation is a research. So some sort of barriers and facilitators can also apply in other research. So this process of reviewing eligible studies yielded 21 barriers and 12 facilitators. We conducted a face and content validity. So face validity was carried out with two faculty members. We asked them to review the language, the clarity of the presentation. We also gave our survey to 2 health economists, one of them working in Saudi Arabia and the other one was working in Kuwait. And we asked them also to read the barriers and check whether if there's any additional barriers that can be added to our list. We also included, one of them was from Kuwait, to check if the terminology used in our survey is consistent with other people in the GCC. Next. Can you move to the next slide, please? Okay, in order to increase the response rate, so one of the limitation of electronic survey, as you all know, is the response rate. So we send weekly reminders to our participants, and also we promised a donation for each completed survey, and we believe this will probably increase the response rate. Next. So out of 143 recruited respondents, only 53 responded to our survey. So this is a response rate of 37%. Next. So this is the demographic of our participants. So as you can see, my previous slide was showing the significant numbers, but I will try to summarize them here. So the vast majority, if you can see, they were young, so from 30 to 44. They were young researchers. The vast majority were Saudis, which is very excellent. 50% of them were have a doctoral degree. Also, they are very young, so the years of experience was around, was up to 10 years. The vast majority of economic evaluations were also conducted in Saudi Arabia. As you can see, so this is consistent with the systematic review findings. So 50% of the respondent published only 1 to 2 economic evaluation. 50% also were academics, and the others were from hospitals and other and Ministry of Health. Next. So in my previous slides, I showed the full 21 barriers and facilitators, but these are the top rated barriers and facilitators. So according to our respondent, they believe that the lack of local effectiveness data and the lack of quality of effectiveness data were the top rated barriers. In addition, they believe that there is lack of financial support, lack of national collected health statistics. So from the barriers, we can see that the access to data was the main barrier, whether it is an effectiveness data, local data, unit cost data. These were all perceived as a very strong barriers. So in terms of facilitators, again, they also highlighted that the availability of data warehouse of all hospitals admissions and outpatient appointments were perceived as the main facilitators. They also believe that establishment of a Center for Health Economics Research will also help. And again, the accessibility of unit cost. And what do we mean by unit cost is the cost of a health service. So the cost of GP visits, the cost of ICU bed, this is not accessible to researchers, and they believe that they need this sort of information to conduct economic evaluation. Next. We also asked our researchers to write or to add additional barriers and facilitators. Some of them said that there is a lack of interest from decision maker, lack of collaboration between national authorities, a lack of local experts, and lack of economic society. They also believe, on the other hand, they believe that the collaboration between health economists and the GCC will also help. Introducing postgraduate programs will also help in enhancing or boosting this this field. Next. So in this discussion, it is clear that the access of data, whether it is cost, effectiveness, unit cost, is the main barriers. And again, there is a clear alignment between the barriers and facilitators, which indicate that the data is the main driver for, or is the main issue in conducting economic evaluation. Next. So this is the brief recommendations for the GCC countries based on international experience. We suggest, or many other countries are using real-world data to conduct economic evaluation instead of conducting RCTs, because RCTs are also associated with cost and many other barriers. So we can use real-world data in economic evaluation. We also suggest that establishing and developing an integrated electronic medical record between private and government hospitals will also help us and capture the full journey of the patient, and it will give us a clear idea of the accurate health resources use. Finally, bridging the gap between researchers and decision makers. This can be achieved by streamlining the collaboration between academics and people from other from national bodies and inviting them to national projects. Finally, we also recommend establishing more health economics postgraduate programs. Next. Next slide, please. So this study, as a conclusion, this study highlights that the data accessibility and availability of data is the main barriers and facilitators as well. Using real-world evidence is a promising solution for economic evaluation. Thank you so much for your attendance.